Professor Nabil Abdul. Professor and head of biochemistry, DMS Punjab University, Chandigarh. Idea of distribution and lipid metabolism, economic and text biology. She has done her PhD from Chandigarh. She is devoted with the I request James our sessions.
Institute of Readiness Foundation. Your reading committee is it is PhD from Boston and several postdoctoral fellowships primarily in USA, University and Director at Incd Pune, Professor of Medicine at the School of Life Nation. He is the and probably get pleasure today to listen to him like many times when I talk to him, he always start with your
Thank you, sir. Lights off करो. Can you have one water bottle? Yeah.
Good morning. I like to thank Professor Alok, Professor Ajay, Professor Subhas to organize this event. I remember I also attended that earlier Incd, one organized in Rishikesh and another one at IIT Guwahati. Of course, I need to thank Professor B.B. Agarwal, who I knew my first interaction with him back in 2003, when I published 5 papers in 1 year, Journal of Biological Chemistry, from National Center for Cell Science. One day I got a mail from Professor B.B. Agarwal that Dr. Kundu, I saw your paper. I need to get a one cDNA construct called LCK, leukocyte kinase. I did not know who was Professor Agarwal. Then I sent it from Pune by post, and then immediately he contacted. He said, thank you. He was trying from other university in US, but when he saw our paper, and then that was my first interaction. And since 2003, Then I was part of his family, Society of Translational Cancer Research, INCD. I was also co-organizer of his third International Conference on Society of Translational Cancer Research, which we organized in Lonavala, I think back in 2007, if I remember. With that, I'll not take much time because we started late. And also, I'll try to finish within 20 minutes. So after coming back India, staying almost 9-10 years, including NIH. So I never worked in the area of cancer when I spent 10 years in US, especially work on cardiovascular research and other area with chemistry background, moving to molecular biology, cell biology, immunology, etc. But after coming back India, that time director said that you have to start something new. So that was my beginning of cancer research journey. So title, as you can see, therapeutic targeting of different subtypes of breast cancer using nutraceutical based approach. I just share some information. This is very unique, and this is how it happened in India. So when I was director in CCS, short span of time, and so I submitted one project along with my team to DBT BIRAC as well as funded by World Bank through National Biopharma Mission Program. It's called NBM. And that objective of that program was establishment of GMP compliant national repository for banking, safe deposit and supply of characterized cells for use in biopharma industry in India. There is no government program so far on this area. So we submitted, I was coordinator of the project, and after multiple review and multiple interaction, project was funded 24.5 crores INR for 5 years. And the purpose of that was that biopharma industry in India, those who are developing druggable molecules, they have to independently test in other facility. Mostly they test outside India, which is very expensive. So the government decided that can we have that kind of such kind of facility in India. But unfortunately, I got this project 2019 beginning, and 2019 November I had to leave NCCS Pune. And after that, next director could not activate this project. Although they received 50% of fund, about 15 crores they received. Till today, the project is not activated. Coming to cancer, many of you know, and then yesterday there's a lot of talk. So the message I like to give, I don't see many students here, although Alok spent huge time and efforts and fund to organize this conference. I was expecting more students should participate in this program. So any nutraceuticals or phytopharmaceuticals, if you want to test it, you need to have a good validation, scientific validation, good target, good model. And today, any cancer, any solid cancer, unless until you understand that tumor heterogeneity, you understand the tumor microenvironment, it is very difficult to know where you are targeting, whether you are targeting in the tumor cells or you are targeting in the stromal cells, which includes cancer-associated fibroblasts, endothelial cells, many immune cells, including macrophages, tumor-activated macrophage, NK cells, T cells, pericytes, and many other cells, including some subsets of cells that is called tumor-initiating cells, TIC, or cancer stem cell. So I'll be talking mostly breast cancer, but I'll show some data related to other cancer as well. So breast cancer, those of you who are working, in general, 4 subtypes: luminal A, where that ERP are positive, HER2 negative, luminal B, all 3 positive, hormone receptor, HER2 overexpressing, 4th one TNBC or basal-like, what I have seen, I will show you slides with my other collaboration. India now, earlier cervical cancer was the #1, but last 5, 6, 7 years, among women, breast cancer becoming #1. And the patients, what I can see with my collaborators lab, Tata Memorial Hospital, Mumbai, as well as my current cancer center, Kolingo Cancer Center, KCC, KIMS, KIT University. Women age group of even before marriage, age group of 27, 29, 30, 31, they are developing triple negative breast cancer, very aggressive, unlike luminal A, luminal B, or HER2 positive breast cancer. The last one, that is called IBC, inflammatory breast cancer, not very common in India, where that entire organ become inflamed. This is how it works. Starts with the normal duct when it becomes carcinogenic, and then this is called ductal carcinoma. And when these cancer cells become more invasive and aggressive, that is called invasive ductal carcinoma. And it happened in that lobule also, that is called lobular carcinoma. Started with a little lump here and there, as you can see, and then tumor grow at the primary site bigger, and initially targets in the lymph node. Lymphatic area is the first target. From there it goes to lung, liver, and bone and brain, like prostate cancer as well. So therefore, understanding that development of the cancer at the primary site as well as metastatic site, especially need to do a lot of work in order to understand that mechanism and progression of metastasis. This is one of the project we are running. Still this project going on, 5 years project. When Professor Harold Verma visited, when Dr. M.K. Vaughan was Secretary Deputy, he is no more. That time he suggested, can India have NCI India, which started in Jhajjar with 3 components, and parallelly DBT can have a VNCI program, DBT VNCI, Virtual National Cancer Institute program, in all different cancer. It's like a NCI spore kind of program. So I was part of that breast cancer VNCI, started around 2017 with 5 institutes: Tata Memorial Hospital, TMH, ACTREC, National Institute of Biomedical Genetics, NIBMG, IOB, Professor Akhilesh Pandey, part of that. And but he moved from Johns Hopkins to Mayo Clinic, Rochester now. So he was not able to continue this project. And I started from NCCS and transferred this project to KIT University. So objective of these projects were established primary culture and cell line from hormone resistance and sensitive breast cancer patients' tissue, and all samples with proper IRB and ethical consent and clearance sample will be collecting from Tata Memorial Hospital, and that's the way we have done it. Second component was my component, along with first component, development of patient-derived xenograft, PDX model. This is unique model. Many countries, they have developed all different kind of cancers and their respective PDX model, but we are always late. So India, I don't see any PDX model so far. Once you develop PDX model, screening of new drugs, testing efficacy of selective known drugs, identifying therapeutically relevant biomarkers in this PDX model. Number 3, identification, validation of novel therapeutic target from either directly from patient or PDX or primary culture using phosphoproteomics, whole genome sequencing, whole exome sequencing, and transcriptome based approach. So this was the collaborative project, and my role was that to collect sample from Tata Memorial Hospital. I started this when I was at NCCS, and now this project continuing 1 more year till 2023. So depending on the size of the sample, some sample we directly implant into the immune deficient mice to develop PDX. This is very time consuming because PDX takes more time than injecting cancer cells. Then you have to generate generation 1 to generation 2, 3, 4, etc. to see that they are consistent, they are showing the same phenotype and same pattern what you see in the patients. If they are luminal positive, they should show that luminal positivity in that generation 3, 4, 5, etc. Some of that we directly processed for establishing primary culture, thereafter establishing cell line, because till today we don't have any India-centric breast cancer cell line available. Depending on the size of the tissue, some of that we process for both PDX and primary culture. These are the sample identification number, patient's ID number. As I said, some used for primary culture and some used for PDX and some combination of both. These are the students left and right, they work in this project, and representative tumor. Since these are luminal positive, so we had to check the status of ER alpha, PR alpha, HER2, along with EMT specific and proliferation specific markers such as ECAD, NCAD, Vimentin, Ki67, pan-cytokeratin, etc. So this is, I'm showing generation 1, patient ID number P014R. Patients admitted Tata Memorial Hospital around 2015-16 with multiple treatment, combination of treatment, and then patient passed away, I think in 2020 or 2021, but we got all the samples. And these are again some representative of different specific PDX, and these are generation 4. As you can see, left side panel A, the representative tumor, panel B, immunohistochemistry, immunofluorescence and confocal data, along with some other signatures and marker, including alpha SMA. They are showing ER positive, they are showing PR positive, that is the final message, HER2 positive, and some of that Western data, panel C. So then we did some differential expression of this gene, comparing patients versus G1, G2. And we found that there are multiple pathway specific, including that cell signaling pathways, MAP kinase pathways, apoptosis pathway specific, metastasis pathway specific genes, they are expressing in this sets of data. Panel A, we are showing representative tumor from generation 1 to generation 5, along with that immunofluorescence and confocal. Panel B, showing that palpability of the tumor and the growth of the tumor. If you can see with the higher generation, time taken to grow the tumor and to become palpable, it's shorter than that generation, earlier generation. And we have done that correlation of gene expression by gene spring, panel C and panel D. And what we can see, they are tightly correlated between that patients as well as generation 1 and higher generation. And based on gene ontology analysis also, we have checked that functional annotation of genes which are differentially expressed between primary tumor and different generation of PDX. So I'll now go to that some other area. We also did some work related to curcumin. Earlier days, we have shown that curcumin not only targeting NF-kappa B, it target also that one of the proteins or genes which work on osteopontin. So it target osteopontin regulated angiogenesis by specifically targeting this transcription factor, including NF-kappa B. It came earlier, cover page of the journal. Recently, 2019, after moving to KIT University, I got one more project through DBT related to curcumin, nanocurcumin formulation, along with drugs like a 5-protosylurea and other drugs targeting breast cancer. So this was like multi-pronged approach for preclinical assessment of novel curcumin nanoformulation as a drug for breast cancer therapy, and testing in that patient-derived primary culture, PDX model, to check their acute and chronic toxicity, their pharmacokinetic study, biocompatibility, drug release, long-term stability. So this is also under mission mode project because DBT started that independent curcumin task force 3-4 years back with Professor G. Padmanabhan as a chairman. So we submitted this project along with my clinicians from KIMS, KIT. We got about 1 crore project. So we generate a lot of data. Maybe next INCD, I'll present this data. Another phytopharmaceuticals related, yesterday other people talked about this androgaphilite. In collaboration with the National Chemical Laboratory, NCL Pune, Dr. Tulasi Ram, who is basically by training chemist, he purified this and characterized by mass spec, SPLC, etc. And then we have tested in our breast cancer model. And we find that androgaphilite suppressing breast cancer growth. As you can see, panel C, D, tumor growth, tumor volume, tumor weight. And this has been done using that breast cancer cells tagging with the luciferase genes, because this is the bioluminescence technology with that IV spectrum model. We have captured this image and quantified this image. And I was the first person in CCS to develop this technology, going to Hokkaido University, Japan for 1 month as a visiting professor, learned this technology, came back. And 2009-10, I started this technology in India first time. I'll switch a little bit to the skin cancer, connecting with this androgaphilite story. Many of you know skin cancer is a major issue, especially Europe, Australia, US, mostly because of mutation of BRAF and NRAS. And major cases, patient die because of lung metastasis. One of my students, Dhiraj, did PhD, then moved to MD Anderson Cancer Center, and then moved to Columbia University. His boss moved to Columbia University. Now he joined as a group leader at Johnson & Johnson. He asked this question that whether cancer stem cell exists in skin cancer model. So we started with that all melanoma skin cancer specific cell, starting with B16, F10, whatever available, mouse specific, F1, human specific, A375, SKML2, 28, with different mutation of BRAF or NRAS, and whatever signatures or marker available to characterize cancer stem cell, we use almost all in this particular model, starting with CD133, 20, 271, ABCG2, VEGFR1, CD166, CD44, etc. We thought, can we have a double positive cancer stem cell? But when we did that by FACS sorting, we find that they are less than 1%, 0.8%. So it's very difficult to, as a graduate student, PhD student, to continue the project because it's a very laborious work. So then we decided to work on CD133 and positive and negative cancer stem cell, and move further and to check first what are the differential expressed genes which are present in CD133 positive versus negative, heat map analysis, volcano plots. And then I'm not showing all details data because this conference is related to nutraceuticals. So then we developed that good lung metastasis model. And then we tested this multiple so we developed lung metastasis model by injecting CD133 positive cancer stem cell and looking at multiple druggable molecule, including androgaphilite, which somebody shown in that clinical trial going on in that colorectal cancer, CRC. So we also tested that. So this is our B16, F10, LUC derived CD133 positive cancer stem cell injected through tail vein, develop a lung mets model. And this is ex vivo image. You can see left one is CD133 positive, right side CD133 negative cancer stem cell. Panel B, you can see that micro and macro mets. And panel C, the level of VEGF expression also gone down in response to androgaphilite with 150 mg per kg body weight. Again, the issue here, this paper published Journal of Investigative Dermatology, which is one of the top journal in the area of dermatology. But we extensively dissected that mechanism, how that CD133 positive cancer stem cell develop lung metastasis. We find that Notch1 is predominantly active in CD133 positive cancer stem cell. We also find that androgaphylite behaving like a gamma secretase inhibitor, like GSI, which blocks that to formation of active notch called NICD1 from Notch1. And then NICD1 ultimately suppress that CD133 dependent MAP kinase activation, including P38 MAPK, and that inhibits that transcription factor AP1 dependent MMP29 and VEGF expression, and thereafter suppression of angiogenesis and metastasis. We have also used multiple drugs like tramitinib, dacarbazine, dabrafenib. Those are the 3 drugs today using for treatment of skin cancer. In combination of drugs or independently with their IC50, etc., we checked with GSI and also checked with androgaphylite, but androgaphylite also suppressing lung metastasis. As I said, this paper published Journal of Investigative Dermatology. Now I'll talk a little bit about, I'll finish maybe within next 5 minutes. This is one of the protein which we working last more than 22 years NCCS and last 3 years KIT University. This is basically chemokine-like protein. This protein interacts with integrin. Some of you will be knowing that this year Lasker Award received by Professor Richard Hines and Eric Ruslauti, based on their major contribution on discovery of integrins. There are many heterodimers, and osteopontin is also one of that ligand interact with alpha V beta 3 integrin, as well as some other integrin, depending on the nature and type of cells. So there are huge international community, they work in this area. There is an International Osteopontin Society. This protein basically has a unique RGD motif, GRGDS, thrombin cleavage site. Basically, this protein once interact with either integrin or CD44, it regulates cell-cell communication, cell matrix interactions, angiogenesis, cell addition, migration, etc. And as I said, internationally, there are many groups. Neil Federko from Johns Hopkins, Larry Fisher from NIDC or NIH, Bob Weinberg, MIT Harvard, and many other groups in Europe, Japan, Australia, work on this. Canada, work on this area. European group shown a couple of years back, circulating osteopontin act as a predictive marker in hepatocellular cancer. They studied 350 patients. One of my students completed PhD with me, moved to Yale University, had a paper in Cancer Cell, shown that osteopontin CD44 axis in row GD12 mediated metastasis suppression. Similarly, osteopontin CD44 also promotes CSC, cancer stem cell phenotype in glioma. Somebody published in Cell Stem Cell. These are very high impact journals. Bob Weinberg published Cell 2008. I'll just come to that paper. As I said, they started with Gordon Research Conference, then FACE SRC every alternative year. And now we have our own international society. From India, I am the only representative. You can see my name there also. This was 2017 conference in Renaissance Baltimore Harbor Hotel, Baltimore. But we do this conference across US. Last 2 years, we could not do it, but again, hopefully next year we'll be doing. Many guys, including Professor Harvey Kenter, very well-known immunologist from Dana-Farber Cancer Center. As I said, Professor Neil Federko, then Professor Susan Rittlings, George Weaver, and many, many others. Amanda Brown, from Johns Hopkins, etc. So targeting osteopontin, and I know my earlier journey, and Professor Agarwal, I was following his series of papers, Cancer Research, JBC, many other journals, Clinical Cancer Research. So what he was doing, that how TNF-alpha regulates NF-kappa B and MMP's expression, activation, and with a series of active compounds from varieties of species, spice. And what we have shown that how osteopontin controls tumor growth, metastasis, and angiogenesis by selectively targeting some of the specific genes which are pro-angiogenic in nature, like cyclooxygenase 2, VEGF, some of the other target like P38-MAPK. Today, if you search it, P38-MAPK is one of the hotspot in triple-negative breast cancer. So you can see that from India, we published series of paper in cancer research back to back, all targeting this area. Bob Weinberg, I told you the paper, what he published, systemic endocrine instigation of indolent tumor growth requires osteopontin. I'm not going to details of this. Final message of this paper is shown that blocking osteopontin's action may yield useful cancer treatment. And so the first author of this paper, Sandra McKester, she is now assistant professor of MIT Harvard. And I had chance to meet Professor Weinberg 2016. And since then, he is sharing some of his reagent from his lab, published large number of paper. He is that pioneer of hallmark of cancer, 2 Cell paper 2000 and 2011. Again, that osteopontin also other nomenclature called small integrin binding ligand N-link glycoprotein, sibling. This is the nomenclature given by Professor Neil Fedorko, Larry Fisher, etc. Basically, osteopontin and other family member of this protein, they call CCN family, like BSP, DMP1. They binds with integrin, but they regulates starting from survival to apoptosis, proliferation, invasion, angiogenesis, metastasis. They published paper in Nature Review Cancer, one of the review article. I was reviewer of this Nature Review Cancer also. Now I'll just show you, yesterday Professor Paul was talking something about that breast cancer model, prostate cancer model. So here what we have done recently, this is very recent studies, how osteopontin transactivates normal fibroblast to myofibroblast or cancer-associated fibroblast, and how that controls breast cancer progression. This is review article by Raghu Kaluri from MD Anderson, Nature Reviews. So, you know, there are many genes, including extracellular matrix protease, chemokines, growth factors. They are involved in the differentiation of fibroblast left side to myofibroblast or activated fibroblast or cancer-associated fibroblast called CAF or TAF. And once they differentiate, many specific genes like stroma-derived factor 1, alpha SMA, SDF, FSP, they start to express. So one of my students, Ramesh, he asked the question whether osteopontin, being a chemokine-like protein, ECM protein, whether regulates this or not. So we started with the human fibroblast cells, and we isolated peripheral normal fibroblast from breast cancer patient, and then exposed with osteopontin, find that yes, it differentiates fibroblast to myofibroblast. Again, I'm not, because of shortage of time, I'm not going through details. What we find that osteopontin, irrespective of interacting with alpha V beta 3 integrin or CD44, it regulates transcription factor called TWIST1. TWIST1 is one of the transcription factor which is highly activated and expressed in cancer-associated fibroblast, along with NF-kappa B and AP1 through ERK and AKT pathway. And that ultimately controls alpha SMA FAP, FSP, especially SDF and alpha SMA, which we have shown by reporter promoter assay, by chief assay, by Western, qPCR, etc. And we have developed two independent tumor model, one carcinogen-induced tumor model, another orthotopic organotropic breast cancer model. By silencing with lenti-based shRNA, I am not showing data. This twist one, etc. also regulates microRNA Incd2022-Hall A, which ultimately controls the feedback AKT through P10 being a tumor suppressor. And this paper recently published on Incd 2021. Ramesh moved to University of Texas Southwestern, Dallas. He started his postdoc. So in this series also now, last 5-6 years, we had series of paper in oncogene, starting 2013, 14, 15, 16, and 18, one more, and 21. And again, that recently we are using some of the CRISPR-Cas9 based approach. Recently paper published in Molecular Cancer. This is a review article, impact factor 41. CRISPR-based therapeutics, a new paradigm in cancer precision medicine, along with some of that paper in Cancer Cell International, who are targeting that immune cells in the tumor, how that IL-6 regulates that STAT3 in that macrophage in the breast tumor model, and some other paper. So therefore, the take-home message, osteopontin is one of that important therapeutic target, and we have published series of review article, including expert opinion therapeutic target, 3 or 4 issues, and 10 cell biology and some others. We are searching for small molecule inhibitor to block the function of osteopontin, because we have done extensive mechanistic studies. Now we are looking for many nutraceuticals and in combination with drugs and with a good model. Good model is very important. If you don't have good model, either preclinical animal model or organoid model, 2D, 3D model, it's very difficult to validate. So we find that osteopontin also targeting androgapillite, targeting osteopontin, it blocks that expression of osteopontin. We are also using some of that siRNA, shRNA targeting osteopontin, neutralizing blocking antibody targeting osteopontin, alpha V beta 3 integrin CD44, in collaboration with Murdoch University, Australia. We are using that Aptamer technology, basically targeting tumor endothelial interactions, angiogenesis, metastasis, and tumor growth at the primary site. Last few slides. With nanomedicine based approach, all of you know that there are many drugs in the market, nanomedicine specific drugs. One of the best example is Abraxane, which is albumin tagged paclitaxel. It is discovered by Abraxane, US based company, Abrax, US based company, and now Biocon and many others, they are making the generic form of this Abraxane. And with that approach, last 10-15 years, we are collaborating with IIT Bombay and many others. We are using chitosan as a biocompatible, biodegradable, US FDA approved nanoparticle. With my chemistry background, we do a lot of conjugation chemistry. Here we have done with that SPDP with the cyclic GRGDS peptide conjugated with chitosan, and then tag with androgapillite or curcumin or other drugs in combination. Here I am showing that androgapillite nano formulation, their size, zeta potential, cellular uptake, and we are using raloxifene as a control, raloxifene nanoformulation versus androgapillite nanoformulation. Both of them independently suppressing tumor growth in breast cancer model, 41 and other cancer model. Their toxicity, hematological, biochemical, they are not toxic in healthy mice. We have shown that, and we have patented. Last slide. some of the translational approach based on our 25 years journey in India, using some anti-PD-1, PD-L1 along with anti-osteopontin as a targeted immune therapy in triple-negative breast cancer. Second approach, drugs like raloxifene and other drugs along with nano formulation targeting in TNBC. Third one, Notch and osteopontin dual inhibitor along with anti-HER2 or anti-estrogen therapy in luminal AB and HER2 positive breast cancer. Fourth one, mTOR and PI3K dual inhibitor along with COX2. Many of you know that like aspirin and COX2 inhibitor now, they are working very well in different cancers through repurposing of drugs mode. So we are using mTOR PI3K along with COX2 inhibitor in our model. And the fourth one in melanoma skin cancer, we are using that dabrafenib, tramitinib along with Notch OP and osteopontin dual inhibitor in melanoma model. As I told you, we are using organoid, soft agar, transwell, memosphere, melanosphere assays, 3D model. We are also using organotropic, orthotopic PDX model. And now through outsourcing, we are studying that drug release, toxicity, PK, PD, ADME, etc., with some selected formulation. With that, 23 students completed PhD degree. Majority of them settled in US. Many of them now assistant associate professor. Currently, my second innings at KIT, 5 PhD students, including couple of students still there at NCCS, 2 postdoc, 3 project student, running 6 equivalent to NIH R01 project, including Indo-German. Many collaboration with University Oslo, Hannover Medical Center, Murdoch University, Curtin University, Louisiana State University, School of Medicine, IIT Bombay, etc. Some of the students here, some other students. Thank you very much.
Professor Shoyek, he's from Division of Molecular Oncology, HMR, NITPR knowledge, and work in
चेंज करो स्क्रीन चेंज हो रही है मेरा I just need a second, sir. Let's see what is your idea. I want to see here only. No. You will get to see, sir. This is the one you. No, this is unrelated. This will appear here first, and then we will see this. Right.
Good morning to all. I am Dr. Suresh Pidau, NTC from the ICMR National Institute of Cancer Prevention and Research. It is located in the Noida. First of all, I thanks to organizing secretary Dr. Alok Chandra Bharati here giving me the opportunity to present my work. Here to explore the molecular signature and establish the drug resistance model in targeted therapy resistance in breast cancer by approaching the omics profiling. As you know that the breast cancer, as earlier speaker, it is already told that the breast cancer now it is rising in the metropolitan cities, and today represents a significant challenge in the public health. So that the global incidence, it shows that it is both excess in the male and the female. So breast cancer is rising. Earlier that the lung cancer, it is topmost. Now the breast cancer is going up, followed by the lung, colorectal, and the prostate cancer. Types of the breast cancer based on the tissue origin and the molecular signature. As you know that the breast cancer, that the invasive ductal and the lobular ductal carcinoma. Invasive ductal carcinoma has comprised many of the approximately 80% of the cases as shown in the invasive ductal carcinoma, and approximately 20 to 20% are showing in the invasive lobular and the other carcinoma. Here the luminal A and the basal A, it is the hormonal negative and the it is the HER2 positive and HER2 negative breast cancer cell lines. So here that the drug discovery, clinical development, approval, and the drug challenge are the continuous process in the cancer management. Latest breakthroughs in the molecular biology and targeted personal therapies have been tailored to the specific pathophysiological condition in different types of the breast cancer. Tumor cells acquire drug resistance against the anti-cancer drug. Here we are lacking the knowledge of the molecular and the cellular events associated with the acquired therapy resistance. However, there is the clinical and the preclinical reports elaborates the different possible mechanisms of therapy resistance. Here it is the so many possible pathways are there. I am only concentrate on the few pathways. So first pathway is the stimulus induction. So that the OCT4, SOX2, NanoG, CIMIC and KLF4 are known stem cell transcriptional factors that involved in the regeneration and the maintenance of the stem cell characteristics. These transcriptional factors also modulate the tumor cells for metastasis and acquired drug resistance in stem cell population enriched in the tumor cells, which generally detected by stem cell markers such as CD44, CD33, EpCAM, gene promoter, aerobic glycolysis process, and suppress the oxidation of TCA cycle for uncontrolled and the proliferation and survival of the hypoxic condition. Overall, the transcription factor involved in the acquiring drug resistance phenotypes and the cancer progression. Epithelial to mesenchymal transition. In normal condition, epithelial cells are tightly interact and tight junction to adherence the junction and support the basal membrane. During the tumorigenesis, cell morphology changed and cells are loosen the basement membrane are disturbed, and finally the cells acquire male characteristics. So E-cadherin, cytokeranin, and some other are the male markers. Various GR, RKT, TGF beta catenin, and notch signaling. EMT induce invasive spreading tendency, stemness character, and promote survival signaling and block apoptosis signal, and finally cells become tumorigenesis and acquire therapy resistance. ROS foundation. Reactive oxygen species generate due to oxidative reaction mostly in mitochondria, and its concentrate in the cell decides whether the cell go to survive or undergo in cell death process via induction death signaling cascade. However, increased ROS level sometimes initiate the transformation of normal cells into cancerous cells and also promote the cell migration. Epigenetic changes in drug-resistant tumor cells. Environmental factors play an important role in tumor initiation, progression, and therapy resistance that modulates the carcinogenic signaling cascade via epigenetic changes. This is the ABC transporter is a very important protein involved in xenobiotic effects, including the anti-cancer drug. It elevates the expression observed in various cancer and tumor recurrence conditions. So P glycoprotein, MDR1 and ABGC2 are commonly used as a marker for therapy resistance in various cancer. It is the post-transcriptional regulation of gene expression is a very crucial phenomena in the modulation of cellular function. The characteristics of cancer cell as well as the drug response are highly dependent on modulation of signal transaction that is modulated by at the post-transcriptional level by mRNA. However, the drug response is vary for different drugs. That's why we are selecting the kinase inhibitor based targeted therapy. In my lab, we developed the drug. In our group, we are focusing the molecular signature and mechanism signaling network, post-transcriptional and post-regulation, regulation, and many other possible events happening in the acquired drug resistance in breast cancer. In continuation of this study, we are looking the new drug target and new drug candidate to resolve the drug resistance challenges. We have developed the drug resistant cells by continuous drug treatment for several passages and evaluate the drug resistance phenotypes at cellular and the molecular level, and then go for the omics profiling. Integrated data analysis and output outcome of the analysis data validated in the clinical samples. Finally, we will try to identify the molecular signature and new drug target in the resistance cells. In my lab, we have developed the drug resistance model in abemaciclib and palbociclib. These are the FDA-approved drugs for the advanced treatment of the breast cancer, and they are target in the CDK4 and CDK6. It is the kinase inhibitor, and we have calculated the IC50 value of the both the cell lines MDA MB231 is the triple negative breast cancer cell lines, and the MCF7 it is the hormonal positive and HER2 negative breast cancer cell lines. Again, we have tested for the urolimus drug because I have recently got the project from the DHR. It is from the DHR funded. So we are working on the urolimus and the rapamycin. It is the mTOR target inhibitor. Ipirubicin, it is from the ICMR project. It is from the ad hoc ICMR project. It is recently got it is after only for the 5 to 6 months back, and the work is ongoing. And unique proteins and mRNA identifications as a molecular signature in abemaciclib, palbociclib resistance model in MCF7 breast cancer. And we found that some of the proteins are upregulated and some of them are downregulated in the abemaciclib resistance cells model in MCF7 breast cancer cell lines, as well as it is some of the proteins are upregulated and downregulated. It is in the palbociclib resistance cells. And some of them are in the upregulated in the one. miRNA and gene identification urolimus resistance in MDA MB231 cells. And we found that the panel of mRNA identified as a potential prognostic marker in the urolimus resistance drug. And mRNA and gene identification in ipirubicin resistance model in the 231 cells, and we found that 11 unique genes identified in the urolimus RMDA 231 cells, and corresponding 31 mRNA identified that targeting the mentioned unique genes. Rather than these outcomes, we have more omics data of drug resistance in analysis process and have to wait to some concrete area. This is my institute. It is the National Institute of Cancer Prevention. It is the ICMR Institute, and this is my team. Without the team, we cannot conduct the research. This is the 2 PhD students, 2 postdocs from the DHR, that the DHR in scientist, and this is a small group. Thank you, sir.
The area of interest includes significant pathways of active metabolism, and the migration, metastasis, and the brain. He studies PhD from School of Medical and Bioethics, CBS. He's one of the young things about the area of it is post-operative fellowship in Sweden and now working at in finding and I trust that people who have to be
Freeze करो. This one, right, sir?
Yes.
Just give me a second more. I'll just share it on Zoom. Okay. Thank you, sir.
Hello. Yeah. Good morning, everyone. First, I would like to thank the organizer of Incd 2022 by giving us this platform to discuss about our science that we are doing in medical oncology department. So again, I'm going to talk about breast cancer, but specifically on triple negative breast cancer, which are really hard to treat. So as already told by two previous speakers, that breast cancer is the most common cancer in India. And if you look in India compared to West, the ER negativity is high. So if you look at triple negative breast cancer cases, which are basically estrogen, progesterone, and HER2 negative, in West it is around 15%, but in India it's like 25 to 35%. And as already discussed by the earlier speakers, breast cancer as such disease is highly heterogeneous. As you can look at here, it can be subdivided into molecular subtypes: luminal A, B, HER2 enriched, basal-like, normal-like, and TNBC falls here in basal-like. Furthermore, in 2017, they did more in TNBCs, and now we have like 6 more subtypes in triple negative breast cancer. We call them basal-like 2, luminal androgen receptor. Then there is like MSL, which is mesenchymal stem-like, IM is immunomodulatory, M is metastatic, and then BL-like 1. So there are different pathways which are basically governing these subtypes. And of course, it's a heterogeneous disease. You might have like MSL and M subtype together. So how to target it? And of course, prognosis is different for each of the subtype. So there are major challenges. As already told by Professor Kundu, that it is, especially TNBC is basically occurs in younger women, less than 40 years. It is highly aggressive than other subtypes, and it is characterized by high relapse and overall survival. It reduces the overall survival. And then 50% patients which turn up to the clinic having TNBC of stage 1 and 3, almost 60 to 70% go to recurrence. So major treatment challenges is we don't have a proven oncogenic drivers for TNBCs. Like in luminal cancer, you have ERPR, so you have hormone therapy for them. For HER2 enriched, you have like Herceptin. For treatment of TNBC, we are mainly relying on chemotherapy. Now, I did my PhD in the area of breast cancer in Wnt signaling, and we were looking like basically in triple negative breast cancer cases, this Wnt beta-catenin signaling is upregulated. So while my postdoc in Sweden, I was working again on breast cancer, but on the non-canonical pathways. And we found that this non-canonical pathways are actually downregulated. So canonical is up, non-canonical are down. And basically this is what we think is happening wrong in triple negative breast cancer cases. So in 2017, we published this paper in OncoTarget. And we show in this paper that aerobic glycolysis play a very important role in triple negative breast cancers. You all know now what aerobic glycolysis is. Basically, these cells, TNBC cells, like oral, most of the solid cancers, they actually consume a lot of glucose, and they actually convert pyruvate to lactate. Not the pyruvate will not go into the Krebs cycle, and this lactate is actually a oncometabolite, and it is actually transforming the cells. So in this paper, we found that there is an enzyme, we all know that phosphofructokinase 1, which is a rate limiting enzyme of glycolysis. An isoform of it, that is PFKP, is overexpressed in breast cancer cells, but not the other two, L and M isoform. And this PFKP basically is driving the aerobic glycolysis in TNBC cells. And we also found that the beta catenin is upstream of this PFKP in the TNBC cells. So when I joined AIMS, so we looked at this PFKP protein expression in patients. So we looked in overall breast cancer, and what we found that the high level of PFKP protein expression is associated with lymph node metastasis, and the PFKP expression is high in ER and PR negative breast cancer patients. We further validated this data in silico. On the left-hand side, you can see that we have looked at the PFKP transcript profiles using Jena Functional Genomics Explorer. Basically, we compared ER positive, ER negative cell lines, and we found that PFKP transcripts are higher in ER negative cells. On the right-hand side, basically, we look into like online metastatic online database, and we found that PFKP protein expression in TNBC cases are associated with bad prognosis. So we know that PFKP is high in ER/PR negative or triple negative cases, and it is driving aerobic glycolysis. So how to inhibit it? First, what we did, we took this 231, these are TNBC cells, we inhibited the PFKP using siRNA, and we found that the important enzyme LDHA, which actually converts pyruvate to lactate, is also going down, affecting the extracellular lactate, as you can see in the graph. Now, till siRNA, it's fine, but we have to remember that this PFKP isoform actually represents phosphofructokinase 1, which is an important metabolic enzyme and is there in the normal cells also. So how to inhibit it? And then we looked at the literature. We were finding like, can we have a natural product that can inhibit it with less toxicity? So we found Q-certin. It's a flavonoid. I will come to it. So we found that Q-certin inhibits beta catenin in TNBC. So we found that PFKP is regulated by beta catenin in TNBC cells. It has also been shown in adipocytes that Q-certin can inhibit PFKP transcripts. And furthermore, in breast cancer, again, it has been shown that Q-certin can inhibit LDHA protein. So we checked with Q-certin whether it can actually inhibit PFKP. So just to tell about what Q-certin is, it's a flavonoid coming from fruits, vegetable, and tea. Mainly, you can get Q-certin in large amount from onions and red apples. Q-certin has been known to be anti-cancerous, anti-inflammatory. So when we treated 231 TNBC cells, there is a downregulation of PFKP at the protein level. You can see at the immunofluorescence level, the green color, that is FITC, which is actually saying about PFKP protein. In treated cells, it's going down. In the lower panel, you can see that when we treated with Q-certin, actually, the LDHA expression also went down. And when we looked at the extracellular lactate levels, just like siRNA of PFKP, Q-certin can do everything. So basically, it is inhibiting PFKP LDHA axis and reducing the lactate. So what does it mean when the lactate is reduced from the microenvironment? Basically, lactate drives migration. And we checked then migration, and we found that 231 cells treated with Q-certin showing less migration compared to control cells. Moreover, the colony formation ability of these 231 cells was also inhibited. So overall, what we say here is that Q-certin is inhibiting PFKP LDHA axis, thereby inhibiting the extracellular lactate, which is actually driving the migration in the chronogenic potential of TNBC cells. The other protein that we are looking in our lab is RNA binding protein HUR, that is human antigen R. It's an RNA binding protein, and it has been shown to have a role in cancer development and progression. So if you look at on the left-hand side, HUR actually, if you look at the protein structure, it has three RNA recognition motifs. And through this, it binds to its target mRNA at the 3' UTR, recognizing the ARE elements, we call as AU repeat elements. And by this, HUR stabilizes its target mRNA. Take this whole complex come out of this nucleus in the cytoplasm. And basically it stabilizes these mRNA, thereby turning the protein translation of these proteins. So most of these mRNA targets are actually oncogenes like HIF, VEGF-A, estrogen, c-MYC, which are regulated by HOOR. And HOOR now have been reported to actually regulate 3,500 mRNA targets. Most of them are oncoprotein. So in this figure, if the HOOR is in cytoplasm, we call it as active. If it is in nuclei, we don't know the function still, so we only consider the HOOR which is there in the cytoplasm. So what we looked into that, we acknowledged that HOOR expression is also associated with the bad prognosis in breast cancer patient, as shown in left, and Q13 can inhibit HOOR by deterring the interaction with its target mRNA. So we looked at the HOOR expression. We published this paper recently in Nutrition and Cancer, and we show here HOOR is mainly localized in the nuclei, but when we treat it with Q13, actually the cytoplasmic localization goes away. So on the right hand side, we have shown in the graphical format that it almost reduces, Q13 reduces the cytoplasmic HOOR to about 60 to 80% into breast cancer cell lines, NDMB231 and 468. We also showed that Q13 has ability to inhibit the migration of breast cancer cells, both the cell lines 231 and 468, and through transfer assay. And we further showed that actually this inhibition of migration is because Q13 is impairing the adhesion of TNBC cells. Then we validated these experiments by using siRNA approach, where we just knocked down HOOR and showed that yes, adhesion and migration is affected, thereby saying that Q13 is actually inhibiting migration invasion through HOOR. Further showed that the two downstream target of HOOR, beta-catenin and CD44, were also reduced when we treated the cells with Q13. And we also explained that actually Q13 is inhibiting HOOR, thereby inhibiting beta-catenin in TNBC cells. But Q13 mediated inhibition of CD44 is not dependent on HOOR or beta-catenin, and might be there is an independent pathway. Now, in my lab, we are working on natural products. Basically, it's our in-house projects. We are not funded by it. So whenever we come something like to look into, we'll use natural products. So while looking at Q13, we were using dose like 40 to 50 micromolars in TNBC cells. So we thought Q13 is inhibiting migration of TNBC cells, which is very important for cancer progression and metastasis. Can we bring this dose down by combining more phytochemicals to it? So this is a recent publication this year where we have combined Q13 with curcumin and berberine. And we found that this triple combination is really effective in inhibiting the EMT as well as the migration of TNBC cells. So here I'm just showing the IC50 of all the three compounds. So what we did is that we actually took this concentration like 1/10, so 10 times down, and combined the drugs and looked at the EMT markers. So here we looked at E-cadherin, N-cadherin, vimentin, beta-catenin, CD44, and MP9. So you can see that mesenchymal markers are going down, but you can also see that in MDMB468, E-cadherin, which is actually an epithelial marker, is also going down. But that is a specialty of these cells. It has been shown that in 468 cells, E-cadherin expression is required for the migration and metastasis, because these cells collectively move. They are not like MDMB231 that they move single. So they move in sheet, they move in clumps. So if you overexpress E-cadherin in these cells, the metastatic rate is high. And if you reduce, you will find less metastasis. So E-cadherin is a driver of oncogenesis in these particular type of cells. As we can see that there is downregulation in the EMT markers, we next look into the migration, and we found that compared to individual treatment on the left-hand side, the triple combination is more effective. We further looked into colony formation ability of 231 cells, and we found that triple combination significantly inhibit the colony formation ability of these metastatic MDMB231 cells. So if you look at the colony formation assay, what you do is actually you treat the cells with the drug and you allow the cells to grow and form a colony. Similar to like what we do in patients, we give the chemotherapy, and it is the residual disease that comes back and is the cause of relapse. So this residual disease, people are now saying it's because these are cancer stem cells, they are resistant to therapy. And basically when you look at colony formation ability, basically you indirectly looking at the cancer stemness. So we also thought maybe if the CFU is decreasing, there is a population of cancer stem cells going down. We looked at the CD44 and 24 population, CD4 positive and 24 negative, and we found that the triple combination is actually significantly inhibiting the stemness. Now, colony formation ability we have shown, we have now go one step up, showing 3D cultures that yes, proliferation is inhibited, as you can see by the relative colony area on the right hand side in the graph. The triple combination is really effective compared to the individual combinations. We further show that because cisplatin resistance is a very common problem in TNBC patients, we further show that this combination, when given with cisplatin, is better than the cisplatin alone and the combination alone in both the cell lines. So this is our conclusion till now, that our data showed that elevated PFKP and HOOR levels are associated with TNBC progression and relapse, and the ability of quercetin to inhibit these oncoproteins provide a rationale for using it as an anti-cancer agent. Furthermore, we put forward this viewpoint on the role of synergistic combination of these three phytochemicals, quercetin, curcumin, and berberine, and their ability to inhibit EMT process, migration, and the invasion of TNBC cells. So further studies are warranted to validate the effect of this combination in vivo in TNBC cell models. So my group is also working on gallbladder cancer and oral cancer, and we are taking this combination to there also. With this, I like to thank my Department of Medical Oncology, my funding sources, and my PhD students. So on the topmost, it's Umar, who carried out PFKP work. Actually, he is working on beta-catenin mediated metabolism in TNBC cells, but I have given like a natural compound to everyone to look this aspect also. The second student is Arundhati. She looked at the HOOR study, and the third one is Akansha. Actually, she's working on gallbladder cancer cells, but she finished this triple combination study this week. Thank you. Yes, sir. Yes, yes, sir. An individual also. Yes. Yes.
You can establish like some selective nanomaterial and chemical conjugate or
Yes. Definitely, sir. It's a good question. Basically, we are looking into collaboration for that. Yeah, because we can't do that. Yes, sir. That we are doing in triple negative breast cancer organoids, patient-derived organoids, and in gallbladder. Yes, yes. Thank you. Yes, sir. Yes. Yeah, actually, I have not showed you the full data. Okay, so what we did is that we were actually looking at like non-canonical Wnt signaling. No, we only limited to glycolysis, where we looked at hexokinase 2 as well as PK. It is possible. Yes, it is possible. It is possible, and so we have a DBT funded project where we are looking at beta catenin mediated metabolism TNBC, where we're looking at both the components, aerobic diatosis as well as oxphos. Thank you.
Share kar.
This area addresses hormone regulation of gene expression, endocrine axis, and these are PhD one and automatic.
अरे screen change करो. Give me, allow me a second. Pointer. Slow down. ये है ना सर? Yes. Change. से navigate करेंगे. Forward arrow से. हाँ, इससे navigate. Pointer option दे रहे हैं. क्या लाइन चेंज करना है? स्टक हो गया है. फ्रीज करना है कि फ्रीज करो. ठीक है. आगे नहीं जा रहा ना. ये है ना पहला. Slideshow.
Okay, good morning, everybody. Am I audible? Yeah. So in keeping with the theme of this meeting, what I've decided to do is speak about a molecule which was isolated about 100 years back, in 1925 to be precise. It turns out that this is a furanoflavonoid and was isolated from what is called the Pongamia oil. Now, Pongamia is, in most of our vernacular languages, is called Karanj, and which is why this particular compound was named Karanjan. Now, if you look at our traditional systems of medicine, Pongamia oil and the different parts of this tree have been utilized for their medicinal uses. However, the traditional knowledge has still not been scrutinized with the modern methods and techniques that we have available today. And which is why I feel that there is still a lot of scope in this area in terms of wanting to unravel different kinds of chemicals and their mechanisms of action, which can have potential health benefits. Now, this compound Karanjan is not as popular as, let's say, curcumin or resveratrol or other furanoflavonoids, but nonetheless, there are investigating groups in India as well as abroad who have significantly contributed to the current understanding of what this molecule does to different kinds of cells. And what I have just summarized in this slide is the current understanding of the cell biological effects of Karanjan. But what motivated us to make Karanjan as a part of our research activity are two publications which I will just briefly discuss. One was a publication which showed that Karanjin has the ability to induce cell cycle arrest and apoptosis. And for that purpose, they use cell lines like HEPG2, HL60, and A549. And I would like you to look at the IC50 values of this compound in these 3 cell lines. It is somewhere in the order of 10 micromolar to around 40 micromolar. Another publication from India itself, it essentially demonstrated the same idea. But what was remarkable about this paper is that it brought all the different kinds of components that we know of and different kinds of phenomena within the ambit of Karanjin's mechanism of action. For example, let's say P53, P21, NF-kappa B, DNA damage, caspase cascades, and whatnot. But here again, I would like you to note something. Say, for instance, in a cell line like some of these cell lines, I don't know whether it is visible over here. The IC50, if you look at it, they are extremely high. And that gets you into trouble because you run into a debate as to whether such high concentrations can be achieved in cells where you want Karanjin to act. So in short, whether these concentrations of Karanjin are pharmacologically relevant. But we have decided to steer clear from this debate, but then go on to ask a simple question as to, for all the cell biology that you know of today with regards to Karanjin, what are their genomic correlates? And this is what we believe has not been addressed in the literature as much as Karanjin's mechanism of action is concerned. So we embarked upon a transcriptomic study to look at Karanjin modulated genes using the MCF7 breast cancer cells. I may want to tell you later why we chose this cell line. And for that purpose, what we did is we used MCF7 cells, concentration set to 10 micromolar, keeping in mind the 10 to 40 micromolar IC50 concentration. And we chose to treat the cells only for a period of 24 hours. Now, what you see on the left, the viability data shows you that at 10 micromolar concentration of Karanjin, which is somewhere over here, it doesn't make any difference in terms of the viable cell count. So what it basically tells us is that whatever transcriptomic data that we collect out of this experiment, our inferences are not going to be kind of influenced by some of the secondary effects or collateral effects of a system which is heavily apoptotic or heavily proliferative in nature. And so this particular data has been published, so that I'm not going to get into the details of this, how RNA-seq experiment has been done. It has been done with all standard methodologies in place. However, I would like to summarize the story, what came out of this RNA-seq experiment. We found out that there are about 730 odd genes which are modulated by Karanjin. Roughly half of them to be upregulated and half of them to be downregulated. And as is the standard practice, we just picked up randomly some of the top modulated genes. And we have been able to show by real-time quantitative PCRs that our RNA-seq data is in agreement with what we see in quantitative PCRs. Now, when we did this experiment, we were anticipating that the kind of gene modulation that you would observe following Karanjin treatment should in principle reflect what is already there in the literature, meaning cell cycle arrest or apoptosis. But then we were in for a big surprise. So when we analyzed the gene expression data for the different kinds of hallmark genes which are enriched upon Karanjin treatment, it came to us as a surprise. I don't think you're able to see this because the table is blocking. Nonetheless, I will go ahead. That one of the hallmark gene sets which was enriched upon Karanjin treatment was the G2M checkpoint genes itself. And we have looked at what the leading edge genes in this category are and went on to validate the qPCR. And what you find is there are some genes like c-MYC, or for instance, I think CDC25B, they are all induced upon Karanjin treatment. Now, the manner in which Karanjin induces these genes goes on to suggest that it is actually causing cell cycle progression rather than cell cycle arrest or apoptosis. So this was surprise number one. The second surprise was that we also found enrichment of a category of genes which can be categorized as estrogen response early. Here again, we have looked at the leading edge genes and went on to validate some of these genes. And as you can see, CD44, STC2, then TI-PARP and TFF1, they are all induced by Karanjin in MCF7 breast cancer cells. Now note that TFF1 is basically a classical estrogen modulated gene.
So whenever we do an experiment in the lab to do estrogen treatments, the only way that we show that your estrogen treatment has worked is by showing induction of TFF1, which is also called the trefoil factor 1 or the PS2. Now taken together, the thing is that we have to know that both MCF7 as well as T47D cells, they are estrogen receptor positive cells, and they do depend on estrogen for their growth and survival. And so therefore, the simultaneous enrichment of G2M checkpoint genes on one hand and estrogen response early genes on the other actually makes a lot of sense, particularly keeping in mind that they are MCF7s are ER alpha positive breast cancer cells. But then how do you reconcile this with other people's data that, okay, it is actually a pro-apoptotic, and even our own 24-hour data where we say that, okay, there is not much is happening as far as the cell viability is concerned. So we asked a question, what will happen if you were to stick to 10 micromolar Karanjin concentration, but then expose the cells for a longer period of time, let's say about 120 hours. And here again, we are surprised. And what we saw here is that in fact, in MCF7 cells, you could actually get the cells to proliferate better at concentration as low as 10 nanomolar. And even in T47D cells, that happens at concentration even lesser, in the order of 1 nanomolar. Now, if you ask where is 10 micromolar concentration? So if you look at the 10 micromolar concentration data, you see that the viability is somewhere in between. It is definitely significantly less than what you see in the control cells. But then let us not forget that the cells have actually grown. This is the red dots are the baseline viability. And nonetheless, the cells have actually grown over a 120-hour period. And so what we believe is that when you collect data on 10 micromolar concentration at 24 hours, you are basically capturing a system which is somewhere in between. It does seem to have a hangover of a proliferative pathway, but then if you were to continue to culture it at higher concentration, you would actually end up seeing cell death. One other interesting thing that we saw is that the Karanjin modulated genes in MCF7 cells overlapped very well with those which are modulated by estrogen itself. And for that matter, the ones which are modulated by tamoxifen. And the color heat maps on the right-hand side will basically convince you that if you look at the commonly modulated genes, the kind of regulation that you see with estrogen and Karanjin can be of both ways. Either both Karanjin and estrogen can modulate a gene in a similar direction or in the opposite direction. And we also found that there seems to be a reasonable correlation between the fold changes that you see following estrogen treatment as compared to what you see following Karanjin treatment. So all these data are compelling evidence to suggest and ask a question, is Karanjin a molecule which can produce partial estrogen-like effects in breast cancer cells? Or to be even more direct, do we have enough evidences to conjecture that Karanjin could actually be binding to estrogen receptor alpha and producing effects at least partially in that manner in breast cancer cells? So what we have done is we have actually embarked upon experiments to really figure out whether Karanjin can physically bind to ER alpha. Till now, we have not been successful, primarily because of the fact that purifying the recombinant ER alpha ligand binding domain has been a real great challenge for us. Every time we use different protocols, it ends up into the pellet fraction. And so we are not able to purify it. But while we keep continuing to work on that direction, it was quite heartening to find out through molecular docking and molecular dynamic simulations that it turns out that Karanjin can bind to the estrogen receptor alpha ligand binding pocket in a manner which is very similar to the way estrogen would bind, particularly if you look at the hydrogen bonding with the residues which are present in the ligand binding pocket, particularly the histidine 524, which seems to be very important as far as the ER alpha ligand binding domain is concerned. But then we started to ask a question, okay, well, we may not be having any biophysical data, but can we do some experiments that would suggest that ER alpha is involved? So for instance, what will Karanjin do to ER alpha? Would it be very similar to what estrogen does to ER alpha? And that's what we started to ask. So one of the things that estrogen does to ER alpha is it increases the turnover of ER alpha protein in MCF7 cells. And it is very typical to observe ER alpha levels going down upon treatment with estrogen. And in fact, that's precisely what we see with Karanjan, if you were to treat MCF7 cells with Karanjan, ER alpha goes down. Then the second question that we asked was that if Karanjan is modulating gene expression via ER alpha, can you neutralize that effect by doing an ER alpha knockdown experiment? So one of my students in the lab has already shown that cystatin A is an ER estrogen signaling target, where estrogen can suppress the expression of cystatin A, and that suppression requires ER alpha to go and bind to the regulatory sequences in the locus of cystatin A. So as you can see over here, what you can see is that Karanjan by itself can downmodulate the expression of cystatin A, but if you were to do an ER alpha knockdown, this downmodulation is lost. Similarly, the downmodulation of ADAMTS19, which is another estrogen suppressed gene, can be mimicked by Karanjan, but the downmodulation can be blocked by ER alpha. I talked to you about the classical estrogen regulated gene TFF1. That we do see an induction of TFF1 with Karanjan, and that induction can yet again be blocked by ER alpha knockdown. And to top it up, we could also demonstrate using chromatin immunoprecipitation assays that while Karanjan is able to modulate the expression of all these estrogen regulated genes, we could also demonstrate that ER alpha actually goes and binds to the estrogen responsive elements in all the 3 estrogen regulated gene loci that I have spoken to you about. So all these results finally get me to conclude that the Karanjan's transcriptomic footprint is much broader than what we expected. The surprise was enrichment of G2M checkpoint genes and estrogen response early genes. While at the same time, we have data that high concentrations of Karanjan can induce cell cycle arrest and apoptosis. What we have also found out that at low concentrations, it can actually promote the proliferation of MCF7 breast cancer cells. And that basically red flags our notion that Karanjan is an anti-cancer or a potential anti-cancer agent. I think we need to look at the concentration dependence far more critically before we are able to draw some conclusions on this. So the question is, where do we go from now here? So in the next couple of years, we would be busy in trying to figure out exactly the opposite of what is being said in the literature. The question is that because Karanjan cannot proliferate the MCF7 cells as good as estrogen, but yet has partial estrogenic activity, could you try and use this compound in something like hormone replacement therapy? So I'll just end this talk with that. Finally, thanking some of the funding agencies. This particular project has been funded by DST Sub, and the last 2 students in this photograph have been the ones who have crafted out these experiments and interpreted the results. I'm happy that Akshita, who has done the entire data analysis for the next generation sequencing, is right now sitting over here with us. And since I think I have saved a little bit of time, I would be happy to answer any questions if you have any. Thank you.
Very nice one. I only one question. What proliferation or acid use?
Yeah, so we began by doing MTT assays, all right? But the problem with MTT assays is that you have to have a whole bunch of 96 well plates to take the 0-hour data, right? Because we want to know from your beginning whether the cells have at all proliferated or not. Because I would only say that a compound is heavily apoptotic if my cell numbers decrease, right? So what we have done is we've chosen to do trypan blue staining assays the hard way out, but probably it has given us much more insights than simply doing your MTTs. No, we have stopped. No. So for critical experiments, we have stopped doing MTT assays in the lab.
I have one question.
Yeah, thanks.
You expose these one.
Okay.
And then use your
No, that's a very interesting question. We have actually not really begun to think in that direction. But some time back, we did think about a challenge experiment. Like say, for example, you have Karanjan, but you challenge with tamoxifen and the vice versa. That will actually give us clues whether they are competing with the estrogen response. I mean, the ER alpha ligand bonding. That definitely insight. Exactly. So we had plans of generating tamoxifen resistant cells, but we have still not been successful. I was given to understand that resistant cells are available with ATCC. So probably we would take that route in an event that we are not able to generate it ourselves.
Breast cancer main problem is ER negative. So what were they?
No, it doesn't do anything to the viability of MDMB231 cells. It doesn't do anything to MDMB468, or sorry, MDMB453, which are also ER negative breast cancer cell lines. So our typical observation is that the ER negative breast cancer cells are not touched. So we do see this phenomenon of inducing the viability or viable cells in MCF7 and T47D cells, but we don't see that in MDMB231. Yeah, 10 micromolar dose. In fact, we have looked at all doses, right from 1 nanomolar to 50 micromolar. Yeah. So in fact, because of time, I have not presented it. We have done the transcriptomic analysis of T47D also. It overlaps with MCF7, but there are differences because these are phenotypically different cell lines, although they are ER alpha positive. Yes. Yeah. But it does overlap, but there are differences too. See, I'll tell you, the moment you cross 40 or 50 micromolar, I'm 100% sure that the cells will apoptose. But the problem is that you run into that argument whether that is pharmacologically relevant concentration or not. Will we ever be able to achieve that high concentration to be able to see anti-cancer effect in vivo? I'm still not sure. So right now we are steering out of that debate and then working out the estrogen receptor angle to the mechanism of action of current gen. Thank you.
Next is Dr. Anna Ghosh. The area of interest to biomarkering certainly no regulation. He has a PhD from the University of Louvain, West Bengal, and he has been a host of the Belgian fellow in Kansas. He has two patents and more than 20 publications.
Allow me a second, sir. Yeah. Just give me a second, sir. Actually, the computer let it slow. Okay. Take it down. Stop here. Stop here.
Refresh. Sorry, there is a technical glitch. Just allow me 5 minutes. Thank you. Sorry. A very good morning. And at the very beginning, I'd like to thank the organizers, especially Anju Madam and Alok Bharti sir, for giving me this opportunity to present my work here. So today I'm going to present a part of my work that I did as my postdoctoral fellowship, as well as what I'm doing in my current lab. And today I'm going to talk a little bit about the regulation of triple negative breast cancer by EGCG, and the entire story will be told with a little twist. Now, firstly, I don't have any conflict of interest in this matter. And so from the beginning of the session, every speaker has told you about breast cancer. And it is very much known that when we talk about breast cancer, the triple negative is the most dire form of breast cancer that we come across. And so I have been working in this field for some times now, and the main issue that came up is basically we want to make breast cancer, triple negative breast cancer, responsive to certain type of hormone therapies. And while doing so, my work is basically focused with a particular group of protein, that is CCN family of protein. Sorry for the bar, it's covering up the heading. And the CCN family of protein is a highly conserved secretory protein. And the beauty of this group is that it has 6 members, and all the members are highly diverse in function. And when I say it's highly diverse in function, it means that if you look at the first member of this group, specifically CCN1, it acts as a tumor promoter for pancreatic cancer. Whereas if you look at the CCN5, another member of the group, it acts as a tumor suppressor for breast cancer. So my work has been concentrated in this particular group of protein, CCN5, and so it's a small size protein, specifically 29 to 35 kb, and this. This slides jumble up. I don't know why. So we did some cell viability assays as well as some apoptosis assays, and we found that EGCG has the property to induce apoptosis and reduce expression of sorry, in the increased expression of CCN5 in breast cancer or TNBC models. So we did some further assays with 3D assays, specifically sphere formation assays. We looked at the size of two separate cell lines, and after treating it with EGCG, and we collected those pairs. We looked at the basic stimulus markers, and all the expression levels were going down. So till this point, we are very happy to see that, okay, there is a molecule that can induce the expression of CCN5 in TNBC cells, and that particular molecule could be used for our purpose. But till that point, we were thinking about why not give it a next level and go for a target therapy with this particular EGCG. So we approached to our chemist collaborators, and we requested them whether we could coat this EGCG with cartesian molecules and attach a PEG and a folic acid along with it. And they did exactly the same. So they basically made two molecules, one with the ECG nanoparticle inside it in the middle, and the folic acid and the PEG was on two separate sides, and the other molecule has a folic acid and PEG attached together with the nanoparticle. So we took these nanoparticles and did our functional experiments with it. And on their part, they did some characterizations with the size of these nanoparticles. Along with those, took some pictures, and they had looked at the amount of drug that those nanoparticles carry. So while the first thing we had to look into is whether after all these modifications and nanoparticle coating, whether it's going through or it's doing its job as a target therapy. So we included this, put this drug into, or this nanoparticle into TNBC cells, incubated them for 48 hours, and 24 hours, and we could see that the nanoparticle weight, particularly that has the folic acid and PEG attached to it on one side, had the highest amount of fluorescence to that particular cell type, a particular compound. So we went forward with this experiment. We did the same thing that we did with our normal compound. So we treated the cells with the same concentration of EGCG. And while doing so, we could see that normal EGCG is basically giving the same effect that has been achieved with the pegylated or folic acid attached EGCG and nanoparticle coated EGCG that we have. Then we did some sphere formation. We did some sphere formation assays as well as some colony formation assay, and on both cases we could find that it is giving us the same result that was being generated as just the chemical without any chemical modification or nanoparticle coating whatsoever. So we took the entire experiment in vivo, and what we saw is basically it's repeating itself as what is seen in vitro setup. And when we use EGCG for treating xenograft tumor models, subcutaneous xenograft tumor models induced with 231 cells, the tumor size went drastically compared to the control. But what surprised us is when we injected the nanoparticles in mice and tried to see whether it is giving us the same kind of result that EGCG is giving. But you could see the second bar is basically the nanoparticle, and it is not giving the desired effect that we have expected for. So that's a setback for this experiment, but in vivo, it is in vivo, so you cannot go do whatever we want and what we expect all the time. So we kept it as an observation, and we are looking forward to work on this particular direction. So in conclusion, what I would say that this particular study has giving us the direction that the EGCG can act as a agent that can promote CCN5 overexpression in TNBC cells, and this particularly nanoparticle coated or nanoparticle conjugated EGCG can act as a future target for used as a targeted therapy for TNBC cells or triple-negative breast cancer in human models. So this work had been published in Pharmacological Research Perspective last year, and these are the mentors that I had from my postdocs, PhD supervisor to all the postdoc supervisors. These two are my just newly recruited 2 PhD scholars, and these are my collaborators that I'm currently trying to work with. That's pretty much it. Thank you very much, and I'll be happy to have some questions.
What are the side effects?
Side effects. So the main thing that when you treat with EGCG is basically at higher concentration, this induces liver damage and as well as kidney damages in normal EGCG drug. When you use it, because EGCG is responsible for inducing antioxidant effects, and too much of EGCG for prolonged time, it induces kidney damage as well as hepatic damages. Yes, sir.
They use combination of recombinant CCN5.
So theoretically, what the last slide I have shown is that when you use just recombinant protein, it has an suppressor, tumor suppressor property too. Now, if you use in combination with EGCG, obviously it will give us a better effect in combination therapy. But the main problem with recombinant CCN5 is obviously to procure it, to make a recombinant protein firsthand. That's the main hurdle, because we have done a lot of experiments with recombinant protein in this particular field, and it's a mess. So I don't want to go to that route in future to treat cells with recombinant proteins, and that's not a viable option though. But that's why we are looking for agents that can induce the level of CCN5 in tumor cells, basically.
I know there are some medical trials.
Yeah. CCN5 patients means no, sir. There is no report so far that looked at that aspect. But yes, there are many trials going on EGCG. Yeah.
This is regarding CCN5. You said that it's a immunosuppressive, this already. So what's the mechanism? Is it mediated or it's something?
It's a ER responsive gene, basically. So,
So we are not going for
no. So yes. Okay, thank you.
Hello. Hello. Hello, this is Professor Rajesh. Am I audible?
अरे उनका आवाज है राजेश साहब.
Hello.
Rajesh sir, please allow us 2 minutes. We are trying to fix the glitch.
Okay, all right. Am I audible?
Sir, we are trying to connect you to the speakers actually. Just
Okay.
allow us 2 minutes.
But I am audible, right?
Yes, you are, sir.
Okay, thank you.
Yes, sir, you are audible now.
Okay, thank you.
Hello.
Hello, should I start?
Lagai.
Hello.
Just
Okay.
a minute.
Okay. Please take your time. No issue.
Hello.
Yeah.
Yeah, you are audible, sir. You can start now. Thank you.
Yeah, thank you so much. Just let me, my screen is full shared or half shared? Is it full now?
Yes, yes.
Hello? Hello?
Yes, sure.
Yeah. So
You are audible, sir.
yeah, I will start. So good morning to all of you. At the outset, let me apologize myself for the inconvenience or to the organizers, which I promised them to be there, but unfortunately, because of some technical difficulties, I could not make it. Today, I will be having a very cursory look at what we have done for this the role of dietary flavonoids in regulation of epigenetic markers, along with those microRNAs which are also involved in epigenetic processes in triple negative breast cancer. I hope my slides are moving. So this is, I just wanted to have a refreshing awareness to the audience that many of may be knowing that these flavonoids is a very interesting biologically active group from the nature, and multiple biological activities have been assigned to this particular scaffolds. Around 7,000 or more than that, even flavonoids have been identified from different resources and have been annotated with different biological functions. Recently, they have also been known to regulate the functions of microRNAs, the circular RNAs, and all those small non-coding RNAs, which per se were not earlier known to have attributed with the functions of these flavonoids. We all know that the recent literature has accumulated, which relates the functions of flavonoids with a lot of cancer cell signaling pathways. They downregulate the proliferation, upregulate the apoptosis process, inhibit the migration, invasion, and also they are recently shown to have some effect in amelioration of emerging drug resistance. And surprisingly, when I was browsing this clinical trials GOV yesterday, there are around 178 clinical trials. People are either trying to use these particular molecules as adjuvant therapy, or they also are interested to link this molecular biological activity of these molecules for circumventing the drug resistance, and to a greater extent also for the off-target toxicities in cancer management. So when we talk about flavonoids and how they regulate the epigenetic frame landscape, that there are some 3-4 aspects that we need to understand. The first is they are known to inhibit these all enzymes which are involved in epigenetic regulation of the cancers. They also have either they activate the tumor suppressor genes or block the suppression of oncogenes. These particular molecules are also reported to repress the non-coding RNAs, which regulate the chromatin structure and aberrant gene expression. And we also note that these molecules also works in methylation and demethylation by inhibition of this DNA methyl transferase and so on and so forth. So it was actually a project given by the SERB, and we set these 4 objectives to work on this. We had a structurally diverse panel of the dietary flavonoids, and we wanted to see whether how these particular flavonoids regulate the microRNAs, either and what sort of differential expression patterns do they exert on the oncogenic and tumor suppressor microRNAs, which per se are related with the functions of epigenetic functions. We also had opportunity to test the combinatorial effect of these the promising flavonoids, which with the standard drugs, and we were interested to know the structural requirements for effective inhibition of these epigenetic regulators by building a 3D QSR model. So this is a framework of our methodology that we have adopted. We first of all kept on screening the flavonoid molecules for the MTT assays, and then Scratch, ROS, that set of parameters that we usually test for the efficacy of any biological molecules that we wanted to ascertain that these molecules have effect on anti-cancer modes. So we have also this panel of HDAC 1 to 11 isomers. We tested all the isomers with this limited test of. We then see how it regulates the onco and tumor suppression. I will let you know. And we also tested some of the ABC efflux transporters, MRP1, PGP, and we also initiated the efforts of cloning the promising tumor suppressor microRNAs in R1 plasmids and see for its functional validations. We have also performed the protein expression profiles by using Western blot, and we did some molecular docking and molecular dynamic simulation for asserting the structure-active relationship. So this is these are my two students, Snehal, which happens to be a PhD student, and Dr. Navnath, who is a postdoctoral, have done this entire work. We initially started with testing the set of flavonoids, some 10 molecules. Actually, we have 10 molecules by in silico screening on various targets of the anti-cancer, and we wanted to ascertain that the test molecules have activity that we have procured from the commercial sources, and we wanted to establish, yes, they do have the biological activity. And we then calculate the IC50 values, and you will also see that some of the impressive molecules like galangine, luteolin, and ricin, camphorol, they have adverse effect on the morphology of tumor negative breast cancer cells. We have used MD-MBA 231 as a model cancer cell line for entire study. We went ahead and we also wanted to see whether these molecules have ability to inhibit the migration, and do they have impressive effect on the inhibition of the migration of the cells. We performed the Scratch assays, and if you look at that, the standard drug which called as SAHA is epigenetic, HDAC inhibitor. As compared to that, there is significant reduction of the wound closure. We know that developing the anti-cancer strategy by inducing the ROS is not a new strategy at all. For example, these drugs, cisplatin, doxorubicin, 2-methoxyestradiol, and these all drugs, they do induce the ROS in the cancer cells and help demonstrate the cytotoxicity and kill the cancer cells. So we have also checked. We have taken some promising molecules like apigenin, galangine, and luteolin, and we see that they do have the ROS inducing capabilities in the triple negative breast cancer cells. You will find that apigenin has demonstrated significant effect in inducing the ROS that we tested by DCFDA dye. We went ahead and also seen some whether these molecules have any effect on the arrest of cell cycle in triple negative breast cancer, and you will find that as compared to the standard drug, they have significantly arrested the cells at G0, G1 stage. We further went ahead and we have seen that how effective they are in inducing apoptosis in the cells, because we know that apoptosis inducing capability happens to be one of the strategies for developing the novel anti-cancer candidate drug molecules. And here you will find that these promising flavonoid molecules have significantly accumulated the number of cells which are undergoing in apoptosis. But we can see in quadrant Q2 that there are number of cells which are undergoing the apoptosis. Then we have selected a panel of 11 HDAC molecules, which our main intention was to see how differentially these flavonoids regulate the expression of these all HDACs, because these HDAC molecules, although they have some consensus sequences in their active site, but they do have different physiological functions. And here you will find that these different molecules have differentially exerted their effects on the regulation of the HDAC molecules. Here you will find that the most important HDACs which are in concert with regulating the progression of the TNBC cancer are HDAC 1, 3, 9, and you will find that here these apigenin and galangine molecules, they do have very impressive effect on down regulating the expression that we have analyzed by using qPCR. You'll also find that these molecules are not effective against DMNT3A, DNA methyltransferase, but they do have effect on DNA methyltransferase 1, which is involved in transfer of methyl groups in the methylation process. We are aware that this ABC efflux transporters is one of the culprit in bestowing the resistance to the cancer cells, because these transporters helps in effluxing the drug molecules and not making available the drugs concentration inside the cells. And we have selected this MRP1, ABCG2, and PGP1 as these three transporters are more explicitly expressed in TNBC cancers. And here you'll find that these three molecules have differential, you just note that the differential regulatory effect of these flavonoids. It's a wrong notion rather that these molecules will have a same effect on regulation of the ABC transporters, but you'll find here that the MRP1 is down-regulated by epigenin, while there is no effect of galangine on MRP1, and same is the case with all other three molecules. Here, interestingly, you'll see that PGP1 is not at all hampered by this apigenin, galangine, and luteolin molecule. So this gives a cues that we need to have some pharmacophore addition to the scaffolds of the flavonoids for the effective inhibition of these transport molecules. We wanted to see as to, we have selected these two representative tumor suppressor miRNAs, MIR334 and MIR200B. The reason that we have selected these MIRs because they are involved and related with a lot of functions of the epigenetic modifications. And we have also selected this oncogene, which is also expressed in triple negative breast cancer, MIR21. And here you will find that these molecules, which are taken further as a promising flavonoids, they have very, very strong effect on inhibition of the tumor suppressors, and they have on the upregulation of the tumor suppressor microRNAs, and they have significantly down-regulated the MIR21, which happens to be an oncogene. We have just initiated the efforts of cloning this MIR124, which is one of the important tumor suppressor MIR in triple negative breast cancer. We have cloned it in R1 vector. Dr. Upasana has done this work, and we initially standardized this protocol. We just wanted to see that whether this MIR21 in cloning stage, when we transfected this particular construct in the MD MBA23 cell ones, you'll find that the MIR124 positive cells, as this particular MIR is cytotoxic, there is less fluorescence of this DS proteins, which is coupled with the CME promoter. And here, in case of R1, where there is no presence of the MIR, which is only a vector, there is a better expression of this DS protein. And obviously, the MIR21 negative and R1 has no expression, as there is no presence of this. So we have also performed these experiments, and we just wanted to confirm that whether this same story happens at proteomic level or no. And here we'll find that the HDAC6, HDAC1, and HDAC3 molecules are being drastically, significantly down-regulated by these apigenin, galangine, and luteolin molecules. We have also confirmed some of the effects of these particular flavonoids on the apoptosis, and you will find that the BCL, which happens to be a pro-apoptotic protein, has been drastically down-regulated in case of, as we all start the apoptosis regulation process with the cleavage of PARP, which happens to be a initial signature for the induction of apoptosis, and here we'll find that apigenin and luteolin has successfully cleared these molecules. At the end, we just wanted to see what happens when we add these molecules which have shown some impressive biological activities. When we take in combinations, we have used this particular SAHA as a standard epigenetic drug, and you'll find that they have demonstrated significant synergistic activity, as it has been calculated by Chow-Tally's equation, where it's a known index that if the combination index is equal to 1, it shows additive effect. If it is less than 1, it is synergism, and here we'll find that the ratio index is less than 1, and here it's clear indication that the flavonoids do value addition in a synergistic mode with the SAHA. That's what we have. In order to unravel the structural active relationship and the molecular interactions with the several species of the HDAC, we have docked these complexes with these flavonoid molecules by using this gold score and scan score, and you will find that they have demonstrated significant scores, and they do have very perfect binding to these active sites of various species of HDAC molecules. We have measured all those HDAC bond angles, the distances, the molecules involved in that, and it's an entire scenario of that. There is a very concrete establishment of all those forces and bonds which per se are required for successful binding of the molecules to the active sites. We have performed the 100 nanosecond molecular dynamic simulation in order to ascertain that what happens actually to the thermodynamic at thermodynamic level, and you'll find that the docked complexes, when they are subjected for molecular dynamic simulation, there is no perturbations, and the RMSD graphs and RMSF plots, which per se indicates the stability of these docked complexes, they are a little bit. You'll also find that here the hydrogen bonding remains consistent up to even the 100 nanosecond simulation. This entire scenario would state that the binding of this apigenin, galangine, and this particular luteolin molecules to HDAC 1 and 3 has sizable comfort zone in binding with the active sites of HDAC 1 and 3. And that has also been shown as what happens, the interactive modes of these molecules when they are under molecular dynamic simulations. So this is, again, we have measured the hydrogen bonding interactions with the simulated complexes with HDAC 1 and 3, as we have done these studies with HDAC 1 and 3 because these two molecules are very instrumental in terms of regulating the progression of TNBC cancer. So just a take-to-home message from our work that these molecules that we have selected for this apigenin, galangine, and luteolin are impressive in terms of showing the biological activities like they are demonstrated cytotoxicity, inhibited migrations, then they have also inhibited the cell cycle. They have induced apoptosis, induced reactive oxygen species, and more precisely, they have inhibited the HDAC 1, 2, 3, 4, 5, 6, and 8. And when we have normalized these results with the GAP-DH. The protein expression levels of HDAC 1, 3, 4, and 6 were also reduced after treatment of these promising flavonoids. And this expression of DNMT1 and HMT downregulated after treatment of these molecules. Interestingly, this oncogenic MIR21 was downregulated after treatment of these two molecules, whereas the MIR34 and 20B was significantly upregulated after treatment of these flavonoid molecules. The combinatorial effect is clearly demonstrated that these flavonoids do exert their effect by synergistic mode. And we wanted to see further, and we have analyzed this, confirmed this effect by this Western blot analysis, and we have also done some molecular docking and MD simulations in order to ascertain the thermodynamic stability of these docked complexes with that. So I would just like to concentrate your attention that the entire story of how dietary molecules exert their effect on in the cancer progression. I have recently wrote this book, which is published by the Springer Nature. This book has taken attention of the Daily Express UK London, and they have given one complete chapter of mine in the Daily Express. I invite you to please have a cursory look at the contents of the book, and some of the chapters may help you to make progress of your research. So these are my kits which we are working here. I had an opportunity to take funding from the SCRB, ICMR, and RUSA. Once again, I apologize myself to the organizers for making inconvenience in the last. I have experience of how difficult it is to have this organization. Thank you so much. Hello. Yeah. If there are a few questions, I'll be happy to take.
Yeah, in short time. So we like to take the
Your voice is not audible to me properly. Hello. Can anybody come on mic and let me know what questions, because the voice is breaking.
So we are very short of time.
Okay. Okay. All right. So should I log out if there is no question?
Yeah, there is a question. Thank you.
Thank you so much. Thank you so much.
Thank you, ma'am. Thank you, sir, for taking the session. Thank you, speakers, for the lecture and the valuable time. I'm requesting the chairpersons to give the momentum to the invited speakers. So I request Professor Mutha and Professor Naveen to request the chair. Now I call upon the first speaker for the day, Professor Gopal. Thank you, sir. Dr. Suresh.
May I log out, ma'am?
Dr. Suresh. Next we have Dr. Chandra Prakash. Next we have Dr. Anil Sivani. Then we have a next speaker, Dr. Arnab Ghosh. And the other speaker, Professor Rajesh Raji.
ओए, इसको जरा ठीक कर.
He will be sending his. You hand over the momentum. Now I request everyone to take a 10-minute break. Just 1 minute, ma'am. I request Professor. I request Professor Agnihotre to present the momentum to the Professor Mutha. Thank you. Take the session of this. And we have session.
ये हरिवाल का इसमें आ गया,
ले लिए पेन ड्राइव?
नहीं, इसमें है, इसमें निकाल दिया मैंने।
अच्छा,
हाँ, नई फोल्डर बना दो यहाँ आगे डेस्कटॉप पे, प्लेनरी स्टेशन पे।
हो गया इंसर्ट? नहीं हुआ?
वो कर दे आगे। अब कर लेगा। ये कंप्लीट हो गया।
हाँ, अब तो सही चल रहा है।
ठीक है,
इंसर्ट हुआ नहीं हुआ? आ गया।
आ गया ना?
कैंडी सेशन 2 के अंदर है ना?
हाँ, किसके किसके आ गए हैं?
लक्ष्मी,
गुरु,
कॉल,
लक्ष्मी आ गए हैं, हरि कॉल आ गए हैं।
तलवार। ये तलवार सर का इनवाइटेड हो गया ना, ये पैनल डिस्कशन तो लास्ट में ना मैंने पेस्ट कर दिया वैसे तो, ठीक है।
हमें अभी किसका चाहिए? गुरु जी की तलवार का चाहिए, हरि कॉल का चाहिए।
कॉल का तो आ गया ना?
हाँ, वो कॉल का मैंने डाला है, और डॉक्टर लक्ष्मी का चाहिए, और
कॉल का, लक्ष्मी का आ गया,
गुरु, हाँ,
और
जी की तलवार फिर का
तलवार, ये 3 का।
ये तीन का आ गया। ठीक है, और इसको मिनिमाइज करके रखो, कॉपी करो जरा फटाक से।
साक्षी
राकेशी और दुलाल पांडे।
दोनों भैया
उनको ऑडियो से
और ऑक्स केवल इसी से जुड़ा रहेगा। ऑक्स केवल
सेशन को टीवी लगा दे होती है तो वो मेला था लगा वो नहीं ऐसी होती कौन से सबको बुलाया है ए टू जेड राइट नंबर लाइट नहीं है तो कैसे कैसे लाइट सिंगर बोला अभी लक्ष्य अरोड़ा का वो जाना संबंधी बन जाती यूं करेंगे तो मार्केट ये कैसे हो जाए हाँ बनाता है ना सबको पता है डॉक्टर जी लगा हुआ ये भाई वो इनको नॉर्मल खाना खाना चाहिए ना गरम खाना अब इसके बाद में मैंने इसको बोला जितना भी यही और दवा एक दिन जो हम लोग खा रहे हम लोग चलो ऑनलाइन में निकलता मतलब श्री राम को रिटर्न बाइबल दे ना पर श्री राम को ना थोड़े बहुत ले जाएगा कैंटीन में काम आदमी का
ये सिंपल था, बहुत सही होता है इसको काम से गरम पानी के एक खाता भी जब से तो सुधर ही आती है
Professor Mehra, I have joined.
जी नमस्कार।
नमस्कार।
आप कौन है? डॉक्टर दुलाल पांडा?
Yes,
Wonderful, nice meeting you.
namaskar. So, is correct session, no? You have started?
Yes, I am on anytime. When the organizers ask us to start off, we will start off.
Okay, so, okay.
Professor Andrew, are you audible?
Yes, you are audible, but your voice was not as good as that of Dr. Dulal Panda.
Can you hear me?
Yes, I can hear you, but there's some amount of echo. So, but anyway, go ahead, please. Go ahead.
All right, so good morning, ladies and gentlemen. Chairperson for the plenary session, Professor S.M. Singh. Professor Incd2022 is Honorary Emeritus Scientist of the Indian Council for Medical Research, New Delhi, India. His areas of interest are infectious and autoimmune diseases, influence of HLM matching and antibody on transplant injury, stem cell biology and regenerative medicine. Professor Mehra has a PhD from All India Institute of Medical Sciences, New Delhi. He is the president of Indian Immunology Society and counselor for the International Union of Immunological Societies. Professor Mehra is founder secretary general of the Federation of Immunological Societies of Asia Oceania. He has received SS Bhatnagar Award of the Society, Ranbaxy Science Foundation Award, OP Hasin Science Foundation Award, Chief of the Army Staff Award, Tata Innovation Fellowship, and the B.R. Ambedkar Award of ICMR for excellence in medical research. The French president conferred Professor Mehra with the title of Chevalier of the National Order of Merit. He is also a recipient of Parsi International Award. We are indeed honored. Our second chairperson his areas of interest are human screening of charts of targeted human associated immunosuppression, post-tumor intervention, tumor gender, stage, and behavior dependent treatment, neuroendocrine prediction of immunotherapy, and Professor Singh has a PhD from the He was awarded Indian National Science and Medal for Young Scientists in 1990. He has received Professor L. Kumar Award. Crystal Research Fellowship from the Japanese Government. Professor Singh is a member of the Indian Cell Biology Association, Indian Academy. He also has papers in national and international journals. For this brief introduction, I now welcome Chairpersons Professor Mehra and Professor Singh to initiate the session.
Thank you very much.
Thank you very much for those very nice introductory words. We like to welcome everybody to this session, plenary session 2 in Hall A. And with me in the chair is Professor S.M. Singh, and I welcome him. This session has 5 speakers, 3 for the plenary talks of 25 minutes each, and 2 are short lectures. And the very first speaker is none other than Professor G.P. Talwar. I don't know whether I should introduce him because he is currently the senior most biomedical scientist of India. And my association with him goes back to early 70s, and rubbed shoulders with him for many decades and learned so much from him. He is a teacher of teachers. That's the only thing that I can say. A remarkable individual who serves as a source of inspiration to a generation of scientists in this country. And I have the privilege of asking him, and first of all, apologizing from him that I am not there in person. I've only joined online. Sir, sorry about that, but I saw you in the hall. You are really remarkable. And we are looking forward to your lecture on a unique vaccine based on Mycobacterium indicus pranae, a potent invocator of immune response for prevention and therapy of TB and leprosy. By the way, let me tell you this MIP, as we call Mycobacterium indicus pranae, actually used to be called in those days as Mycobacterium W, and then Mycobacterium, of course, indicus is the name from India, and pranae. Pran is his name. We rarely call him GP Talwar. We always call him Pran Talwar. And he worked on this on this particular bacillus first in AIIMS, but more importantly when he was the director of the National Institute of Immunology, and that is why the word pranai comes from. So we're going to hear this remarkable story about MIP. Thank you very much, sir. The floor is yours, and the organizers have given 25 minutes for your talk.
I will request Sandy which
Anyway, the subject that I'm going to speak, you will need, they say they need some time to fix up the slides, but well, as you see, this is not the only one area that I work on. I work on the recombinant vaccine against HCD as written there, but my current interest is on immunotherapy of drugs-resistant advanced stage cancers, for which that at present nothing can be done. But with the 4 scholars who work with me, we have reached a point where there may be a hope of doing something for such cases, and hopefully clinical trials will start on it in course of time. to prove what we have found in the laboratory. Now, this particular vaccine, which is, as they say, is also named after me because it is Mycobacterium indicus, India. PRAN, NII is the Institute, National Institute of Immunology, from where the trials on this vaccine were held. And I think it's a very unique vaccine. It has multiple applications. Even for COVID, you know, there were 2 trials carried out. There was no connection at all of MIP with COVID, but still, you know, one trial at AIIMS, another in Bombay, and in both of them, the results were not discouraging. But this particular vaccine was developed for leprosy. Why? Because, may I have the next slide, please? You see, in 1970, Dr. Howard Goodman and 3 or 4 other brilliant immunologists of the world came to see me and asked me to take over the headship of the WHO Research and Training Center in Immunology for India and Southeast Asia.
May I request the organizers to show the second slide, please?
Doing good work, and it's why change at this stage? And I'm quite happy.
Please go to the next
They
slide.
told me that, look, India has the world's largest number of lepers. Do you expect Americans to come and solve your problems? When I heard that, I felt so ashamed. And I took no time in signing whatever they were offering, with the hope that I will do something for which we should be ashamed, that we have the largest number of lepers. Next slide, please. Now you see it isn't that M. leprae is a resident of only India. In fact, it was discovered in Norway. And so India is not the place where this vaccine stays. It was everywhere in the world. But how come that we have done not enough to eliminate, if possible, leprosy from our country? Next slide. Now, you know what I want to say is that, you know, the very first thing in order to do something in a disease is to learn why it is caused. And why everybody doesn't get it, only a few get it. So what's the sort of mechanism by which this bacteria invades and makes a man a patient? So to do all that, one had to learn the sort of characteristics of this bacteria. You know, we've had to first develop a system to grow this bacteria. If we have to find a mechanism to stop that growth. So we developed with Krishnan, who was at that time my student, we developed a system where macrophages made from monocytes, blood monocytes, fresh, could be invaded with M. leprae, and they would incorporate thymidine into DNA. Now, this experiment which I'm showing you has 2 purposes. First purpose is that there is a form of leprosy which is called lepromatous leprosy, which is the multibacillary leprosy, the bad type of leprosy. And then there is a tuberculoid leprosy where it is a limited growth of the bacteria. So the very first question was, why here it's limited, whereas there it is unlimited? Sorry. क्यों भाई? कि कोई ज्यादा है वहाँ? यहाँ मैं अपना स्लाइड तो पढ़ सकूंगा.
Right.
क्यों भाई, कोई बुरा लगता है अगर मैं यहाँ खड़ा रहूं? ये पता नहीं क्यों चाहती हैं कि वहां खड़े हो जाएं फोरम. Sorry. Oh, you mean to say there is a mic there? Is that the reason?
That is a fixed one.
ठीक है यार, यही काफी है. अगर ये अच्छा नहीं तो और दे. So here you see that in the tuberculoid leprosy, you know, there is in fact a low incorporation of thymidine, and more so if you put in the lymphocytes also. So lymphocytes carry the cell-mediated immunity process. So what it shows is that indeed, if you would have an immune response of the cell-mediated type, you can limit the incorporation of thymidine into DNA. Next slide, please. Next slide, please. You see, in order to develop a vaccine, we looked for all mycobacteria which were available, if possible, non-pathogenic mycobacteria, and we named them. So this particular one was coded as MW at the time that we worked on it. And you see, what we tried to find out was what is wrong in patients, in persons who get leprosy, victims become victims of leprosy. What is wrong in their immune system? So ultimately, all that work is published in fact, it's a special Golden Jubilee volume of leprosy in India. And what we could find is that, you see, we were looking for a non-pathogenic but cross-reactive mycobacteria, which could elicit immune response in not only tuberculoid leprosy patients, but also lepromatous leprosy patients, which are, in fact, you know, very stubborn, so to say, as far as the infection was concerned. So next slide. And the defect is the inability of the individual, his immune system, to react against M. leprae, a critical protein antigens of the M. leprae. All this work, which I'm not going into details because it's a long time since it was done, and it is all available to those who want to see it in published form. So we could find a bacteria which was MW, coded as MW, and which could sort of overcome the difficulties in a autoclave form. That was very important because this way we can make it into a vaccine. You know, it will be a non-pathogenic. If you are killed it, then your toxicology studies will be shortened. It won't be as long as, you know, toxicology sometimes takes years and years and years. So you could cut that if supposing you're autoclaved, which is where you are killing the bacteria, that could work as the agent for vaccine. So the next slide. This is what Mycobacterium indicus prani is. Isn't it a lovely bacillus? It's indeed a very impressive, so to say, bacillus, and it has several very good properties, as you will see shortly. Next slide. Next slide, please. यूं जरा अगला चला दो. स्लाइड नहीं रुक आगे चला सकते भाई. MIP, oh sorry, अरे भाई क्या हो गया? This MIC has been passed on to Cadila, so that it would be available to the public. But let me tell you, Cadila has betrayed me. What it does is, when we licensed it, we kept it a condition that it will not be priced very high. After all, leprosy is for poor people. But you know, they found so many other properties of this bacillus. They sell it under different names, the same bacillus, for psoriasis, for cancers, and this and that. And therefore, you know, the availability of the bacillus for leprosy is kept very low. And everybody, whether it is the Indian Association of Leprologists or the many organizations, are come to me to blame, भाई हमें दिलवाओ थोड़े से MIP. मैं कहूँ कैसे दिलवाऊँ, वो पट्टा, you know, he मुझे इनकार नहीं करता, and he will send it a few doses to me. But you know, into the public, they make money more selling it for other purposes. And that also shows that there are several very good properties of this bacillus. It's a potent invigorator of immune response in the individual, in the human body, and it has action on many fronts. Next slide. Now you see, if supposing for leprosy patients, if you give the drugs you have to give, otherwise the DCGI will not give you permission to conduct the trials, but you can add in addition anything else that you want to test. So we combined immunotherapy with chemotherapy, so to say. And shortens recovery time, clears granulomas, and then you know the leptomine conversion. Normally, leptomine is a test, and most lepromatous people are, in fact, one of their characteristics is that they are leptomine negative. They cannot elicit a CMI response against M. leprae. Many of them become positive when they are given this bacillus. And then it is responder and slower. Next slide. See what it does. Here is a case initially of leprosy. After 4 doses of the vaccine, and then after 1 year, look how she clears up. You cannot even imagine that she ever had leprosy. Absolutely clear like any individual. Next slide. These drugs don't do. You know that even if you cure them, they carry their blemishes. Another patient, all cleared. Next slide. And you know, they all came to touch my feet as if I have done them wonders. Look at the back of this fellow initially, and then after 2 doses, his back is totally clear, as if he never had leprosy. Next slide. Yet another such case. with odd blemishes to a very clean back when he had received also MW or MIP. Next slide. See, this fellow was from Nepal and he said, "Oh ji, meri to zindagi bana di aapne. मैं तो कहीं नहीं का जिंदगी में था नहीं था अब मैं कम से कम आदमी तो बन गया हूँ." This is what this fellow said. Next slide. Next slide. You know, in fact, you can have many, all the patients that received the vaccine, in addition to the drugs, became so clear that it's remarkable. You know, nowhere, no drug could have achieved that, what the vaccine helped them achieve. Next slide. Now, you know, despite the fact that this vaccine is available, you know, the government has not taken it up. The government of India, I'm saying. Of course, some state governments have taken up, and I'll tell you about Gujarat. But, you know, many governments have not taken up. And since 2007, you know, the incidence or the prevalence of leprosy is not diminishing. There are still cases and they are increasing, in fact, than what they were earlier. Although the drugs are being given, but at the level that the program is conducted, they don't use the vaccine along. And so they are, I mean, it still persists. Next slide. And the government of Gujarat invited me and also some ministers who at Rajkot in 2017, they decided to adopt this vaccine for elimination of leprosy. Now, the strategy was that, of course, the patients will receive the drugs and the MIP, but the family members and contacts of the patient will receive 2 doses of MIP at 6 months interval. This was and this will prevent them from becoming victims of leprosy. Indeed, in this area, the leprosy has diminished or at least is not detectable. So therefore, you see, there is a potential in this vaccine to eradicate leprosy, this ugly disease from India, provided we take up such program at the national level rather than only at the state level. Next slide. Next slide, please. Now I come to something which we didn't expect. This vaccine is not only good for leprosy, it is also good for tuberculosis. And category 2 type of tuberculosis is difficult to treat. So there has been trials where the MW, this vaccine, plus MDT was given to 48 out of 49 cases. The cure rate is high. Whereas if you give only MDT, only 21 out of the 27 cases were sort of cured. So therefore, there is a potential of this vaccine for this disease, which is, I think, one of the largest disease in the country. Next slide. Next slide, ji. Kya ho gaya bhai? Oh yeah. Look at this. This is the relapse. If you give the drugs the common drugs, the relapse rate after 24 months is 50% in these cases which were tried, which were on trial in this particular project. On the other hand, if you had combined MIP, the light blue is the relapse rate. So relapse rate is curtailed if this vaccine was used for treatment of difficult to treat category 2 tuberculosis. Next slide. Next slide, please. You see Dr. Sumesh Gupta at the All India Institute of Medical Sciences has very ugly patients with anogenital warts. And next slide will show you how this vaccine, can you imagine? It was made for leprosy, cures these ugly anogenital warts from upper to lower. You see how, what a change that has happened. Next slide. Next slide, please. Next slide, yeah. See the anogenital wart, very ugly, and it has completely disappeared. Next slide. Next slide, Kar lo bhai, haan. Look at the feet. Patient and with MIP, how clean his feet become, nobody can ever imagine. This is happening at the All India Institute of Medical Sciences. Sumesh Gupta is the professor who is doing it. Next slide. Now, another very interesting thing was at the Indian Institute of Science, Bangalore, they tried it for myelomas. And look what even that, the cancer is curtailed, so to say, in the type of experiments that they did with different doses. Next slide, please. There are two trials. The lower one was done at the All India Institute of Medical Sciences. There's the other one in Bombay. And both of them are published. And I mean, who expected this particular vaccine to also be useful for COVID? In both trials, the results are, so say, good results. I mean, not that the vaccine was made for it, but it's positive. Next slide. Therefore, to sum up then, this particular vaccine, which I would call it marvelous, look at the range of properties it has. It's based on a non-pathogenic cultivable mycobacteria. Its gene sequence is now known, and it is named as Mycobacterium indicus pranae. It expedites bacterial clearance of granulomas, upgrades CMI responsible of both BL and LL patient. In fact, makes them normal human beings. And then it upgrades immune status to react to fresh challenge with M. leprae because many of them become lepromine positive. Next slide. It is also useful in difficult to treat tuberculosis patients. In fact, in all patients, it should be tried. And then it heals ugly warts of anogenital regions. And then it has preventive and therapeutic action against myelomas and is a potent invigorator of immune response. In fact, I'm using it as an adjuvant in the vaccine, new vaccine that I've developed against HCD. It's a recombinant vaccine, and there I'm using MIP as an adjuvant. So, next slide, please. The credits must be given to the people who worked. And these are my students and co-workers in the work that I have presented to you. So thank you very much.
Thank you very much, sir, for that masterly talk.
Thank you very much, sir.
And if there is anything you would like to ask me, please
Session is
feel
open
free.
for questions.
Feel
After
free.
the wonderful talk of Professor Talwar.
Feel free to ask whatever
I
you
would,
want.
as the chairman of this session, would like to make a couple of comments because I have been a witness to all this that has been shown.
Do you have any idea this non-pathogenic mycobacterium, the one you developed the vaccine, in animal model? I'm sure in humans it is impossible to test this experiment, but when you infect, what exactly is nothing is happening for the mice, sir, particularly? No, the non-pathogenic.
Sorry?
The mycobacterium that is you have used, it is non-pathogenic, fantastic. So in animal model, particularly the mammal model, when it is, I'm sure when it is when
model for what?
Just mice, the regular mice or a rat. When you infect this with the non-pathogenic in human non-pathogenic mycobacterium, do you have any idea, what exactly any kind of phenotypes do you see? Yep, I'm done.
Any idea why is
No,
it
sir.
causes the effect that it does?
When the non-pathogenic mycobacterium is infected with any mammal, so are there any phenotypes? Do you see any phenotypes related to anything?
Phenotypes.
Phenotypes means the physical expression of this infection of the mycobacterium.
So it is used as a killed vaccine. Uske phenotype kahan banenge?
Not the vaccine, sir. I'm not saying the vaccine. This
The
is
live bacterium you want to say.
My
You want
question
to
is
say the live bacterium?
Exactly.
If that is administered
Exactly.
to experimental animals, is there any effect or not?
Is there any phenotype?
No contradictory effect. If at all, these are all positive effects.
Okay, yeah. Thank you.
Thank you.
So far,
Thank you.
nothing wrong has been noticed about this vaccine.
Thank you.
And I'm very proud of that.
No, that's okay. The
Could have
speaker
been.
has asked a question, so that's okay.
Immunomodulatory effect.
Yeah, it's a potent invigorator of immune response. Immune response is against genital warts, against XYZ tuberculosis, and even against COVID. 2 trials showed good results. So just imagine there is no relationship. All that it does is that in addition to the enzyme, to the antigen sharing actions, cross reactions, it is invigorating the immune response in the anogenital warts. See, that's why how they are all being cleared. Very ugly warts.
Yeah.
And at All India Institute of Medical Sciences. So it's one of the best institutes, best experiment, best results.
Organizers, can
Any
you
other
hear me?
questions?
Organizers,
If
can
there
you
are no
hear me?
questions, thank you very much, Professor Talwar, for the wonderful talk. Thanks once again. Thank you.
I fell 16 stairs and I became a little bit, difficult, but I'm getting back on my legs.
That's great, sir. Great. Thanks to all my.
Professor Narendra, you are audible.
Am I audible?
Most welcome.
Am I audible?
I feel blessed actually. It's a great honor for me.
Yes,
Thanks to
you are
Alok
audible.
with this
Thank you so
opportunity.
much. I just wanted to pay
chair
my
this session.
respects to Dr. Talwar for that wonderful lecture. As I said, I was saying that I have been a witness to this whole long story, right from AIMS in the early 70s to what we have seen now. It's a marvelous vaccine that he talked about. and discovered by this master. I also appreciate the question that came up from the audience. It's a very good question, but we will handle it some other time. Thank you very much, sir, for that very masterly talk. And thank you, Dr. S.M. Singh, for giving me a hand to come down this stage. We need to go to the next speaker. Our next speaker is Professor Hari Kaul. He is from the London School of Medicine and Hygiene. Is it London School? Is it LSU HSC in New Orleans, Los Angeles? And he's going to talk about the novel target in oncogene-driven tumors. The biodata of Professor Kaul is right on the screen. He's the head of GU Oncology Research and Associate Director at LSU LCMC Cancer Center in New Orleans. Professor Kaul, please go ahead.
Thank you so much for the kind introduction. I think the slides, you can see them. So, I mean, this is a tough act to follow. How do you speak? Dr. Talwar gave you a life's worth of information, and people still have questions about how do live bacteria cause changes in the mouse. I'm speechless whether I can even talk now after this speech, but it's a tough act to follow. I would like to move your attention back to the basics. We will talk about EIF4G1 as a new potential target for oncogene-driven tumors. So I have no commercial disclosures. In the basics of protein, the central dogma of the protein synthesis, you have a genomic signal in the DNA, which gets translated into the proteins, and the proteins perform the function. We have translational control, transcriptional control. If you have been in this meeting for the last couple of days, you have seen how gene expression change, thousands of genes change, but all the transcribed messages are not translated into protein. There's a regular control of that process. So oncogene addiction in cancer. Can you turn something off here? Need the echo back. The speaker volume. Turn your volume. So I was hearing my own echo here. Sorry about that. So oncogene addiction in cancer is well recognized. There are in tumors, you have multiple gain of function or loss of function mutations, as a result of which are considered to contribute to tumor genicity. Yet, a single activated oncogenic pathway or an oncogenic protein can drive the tumors in a majority of the tumor cases. And all these malignant properties are driven by just one single oncogene. Case in point is mutated RAS in pancreatic cancers. You have P53 mutations which can drive the whole process. But this concept of oncogene dependency has been invigorated and has been tested in many genetic mouse models, as well as in many of the xenograft models where the cells have been mutated, and this is well accepted. But most of these pathways remain undruggable. There are multiple of these genes that are changed, and having the drug targets for them has become difficult. So transcriptional regulation has been studied at multiple levels. We have a special transcriptional regulation, but the translational control of oncogenic process remains still to be understood very well. So what is translation? It is taking the simple messages that cell makes and converting it into the proteins. So you know that one of the proteins that is well studied in this regard is EIF4E, which is a target of mTOR signaling. And EIF4E is bound to 4EBP, which keeps it in the cytosol and does not let it to translate messages. But then once 4EBP gets phosphorylated, this complex gets formed, which brings in together the central component of this translational complex is EIF4G, which is nothing but a scaffold protein, which brings in EIF4E message along closer to the 4A, which helps to bring this whole complex together, and then poly-A binding protein forms the whole complex. Now, one would say, why is this needed even at all? So why is a cap and tail in a messenger RNA very important? Because it is required for regulating nuclear export of the messages. It helps in prevention of degradation of exonucleases, and also promotes translation in multiple other ways. The poly-A tail is required to prevent RNA from enzymatic degradation. It also aids in transcription and termination and export of mRNA from the nucleus. Almost all eukaryotic mRNAs are called mRNAs because they have a poly-A tail. You use poly-CDT to isolate them. Half-life of mRNA has been calculated by multiple methods. And if a message is without a cap and a tail, it has about half-life of 31 minutes. And without a cap, but with the poly-A tail, half-life is 44 minutes. Without a tail, but with a cap only, it is 53 minutes. But if both cap and tail are present, is about 100 minutes. It doesn't make much difference, 30 minutes or 100 minutes. But if you look at how many protein moieties or how many polypeptides are made out of a single message, from 2,900 to 1.3 million. So this is very, very crucial because oncogenes are required in very high quantity. So if you have ability to interfere this process, especially if you look in RBCs, these messages can last as long as 120 days by this process. So cancer cells have taken advantage and have hijacked this process to have their high level of addictive oncogenes for themselves. So this is a structure kind of the mouse is difficult to handle here, but I will try still. So EIF4G is the scaffold protein which brings in poly-A binding protein on one side and 4E on other side with this message, and then ribosomes get attached to it, so it helps load the messages to ribosomes. There is also a cap independent part of the translational machinery, which is an iris mediated, where everything is same except that you don't need 4E to bring messages to the ribosomes. In terms of the translational control from the signaling pathways, I alluded to it before that this 4EBP, which is bound to 4E, keeps this message from coming into the complex. And when the mTOR is active, it phosphorylates this 4BP, and this 4EBP falls off. So you get 4E message to this complex and translation takes place. Yet, many of the inhibitors of the TOR pathway have not proved to be effective in preventing translation, suggesting that there is this phosphorylation independent way of activating transcriptional regulation here. So there are other 4G has 3 isoforms, G1, G2, and G3. G1 is predominantly overexpressed in the tumors. G2 plays part in non-cap dependent translation, and G3 is present in multiple other tissues and plays part in cap dependent translation. So why target then translational control? So as I told you that most of these pathways that are crucial for cancer and developmental progression require translational control, and specifically targeting the translation of specific proteins. How does a cell decide, cancer cell decide, I'm going to only translate these few messages and leave others out? So the selection bias is again handled by this translational control. So we looked at one of the proteins, we looked at all the proteins in the translational machinery, and we observed that EIF4G1 was the only protein which is selectively increased in prostate cancer. And especially during prostate cancer progression, there's a protein level is very high. You can look in clinical cohorts. We looked at TCGA data and other metastatic prostate cancer data. the prostate cancer, neuroendocrine prostate cancer. In each case, we found both amplification and overexpression of this EIF4G1. And also, we found that patients who had high EIF4G1 had a poor survival as compared to the other patients. Then we thought, well, that is enough. Let's look at all other tumor cells. So we looked at multiple tumor biopsies and tumor tissues in the tumor microarray, and we find almost every solid tumor has upregulation or increased expression of EIF4G1. And this is the message level, again, unbiased databases which have been generated by millions of dollars by public funding. If you scan through them, you can find in almost every solid tumor, there is multiple fold upregulation of the EIF4G1 protein. It is not only present here, but you look at survival across all these tumors, high levels of this EIF4G1 protein are associated with poor survival. In each case, not a single case that we could find in multiple tumor types. We also looked at other ways of looking at through the cell lines and each tumors, why this is up, is it the message is up, or there's a fusion of the genes, and in all cases, there are multiple mechanisms that result in upregulation of EIF4G1. We looked at all cancer cell line data and find that all cell lines that are in the cancer genome database, we find there's an upregulation of EIF4G1. We also looked at the protein work, and we published this work a few years back. In 2019, we published, and we, based on these data, we hypothesized that EIF4G1 is potential target for therapeutic intervention. 2 years later, there was another paper from Gerhard Wagner's paper, which did the deep computational analysis with all the powers they had at Harvard, and they came up to the exactly same conclusion. Of all the proteins that you could look, the EIF4G1 is the one of the strongest candidates for therapeutic intervention. So how we study it in the lab, going to the basics, we look at effects in protein translation. You look at how much message is on polysomes versus monosomes, and then you try to interpret these data by looking at either single messages by PCR, or you can do the sequencing of these, which is called polysome profiling. And then you isolate these polysomes from monosomes by using differential centrifugation, and techniques are well established, nothing new in there. So when we knocked down EIF4G1, we found that although the polysome loading was impaired in many cases, but there were still some messages that were being translated. But in each case, shown here is data from 2 different cell lines, that the loading of messages in these tumor cells was reduced when EIF4G1 was inhibited. So was the characteristic of cell growth, colony formation, cell migration, and invasion. Features that we look at were in they were affected by or impacted by EIF4G1 knockdown. 2 of the proteins that we looked at that time point were Incd2022 and SNAIL. Both are associated with transition to the mesenchymal cells or mesenchymal behavior. We found that those proteins were also decreased in their expression. We hypothesized that in normal cells, EIF4G1 drives this unique pathway of cap-dependent translation, but as the tumor progresses, because EIF4G levels are highly upregulated, they drive these processes of proliferation, clonogenicity, cell cycle progression, EMT, and stemness, and therefore maybe promoting cap-dependent translation, which causes therapy resistance. Well, it is not as simple as that. It was a simple cartoon. This is from Trends in Cancer. People, when they look at the literature, they find that there are more than this cap-dependent translation can be affected because the protein synthesis is inhibited due to energy supply, and EIF ternary complexes may not be stable under those conditions. There are the pathways that have been used and the drugs that have been used to target metformin, platinum, Incd2022, rapamycin, and starvation simply to affect the mTOR complex, yet the ternary complexes are still formed and you still get translation of the proteins. In terms of summarizing the Trends in Cancer paper, they said that energy supply decreases as the tumors go from normal cell to the carcinoma and CTO to the highly invasive tumors. And this is because there's a plasticity of the cells is increased, and there's a translational reprogramming from this normal cap-dependent translation to only stress-accepting factors being selectively adapted. So we went to test again. We took the cell lines which were therapy resistant, which is an enzalutamide resistant prostate cancer, and wanted to test whether EIF4G1 still played role under these conditions. We find that indeed elevated levels of EIF4G1 are present in these enzalutamide resistant cells. And if you block it, you again can inhibit their clonogenic activity as well as their proliferation rate and migration and invasion. And a small molecule which Wagner had developed, EI4EG1, again, it is not really a specific and selective molecule, it's a dirty inhibitor, but it does work. We observed that it was not really inhibitor of EI4G1, but it was actually decreasing the levels of this protein of EI4G1 itself. And so this EI4G1 decrease was sufficient to limit the metastatic as well as the tumorigenic properties in therapy-resistant cancer. Again, are shown some more lab data on clonogenic activity, migration, invasion of these cells. So in vitro, it does work very well in these assays, including the sphere formation. Then we also did in vivo assays where we took enzalutamide-resistant prostate cancer cells alone, or with the enzalutamide alone, or with enzalutamide and this drug combination. As Dr. Talwar told you, in patients, you cannot just give a new drug. You have to also give the standard of therapy, which is enzalutamide, in prostate cancer patients. And we can see clearly that you can reduce these tumor growth from the resistant disease just by targeting this pathway. And some more data on the deep sequence data where you could look at multiple genes that could be affected from this pathway and the sphere formation, not only in prostate cancer now, therapy-resistant prostate cancer, but also some data we show you here for the pancreatic cancer cell lines, where you can have impact on the tumor formation in these models. So irrespective of it, whether there is a normal cap-dependent translation process or a reprogrammed process, we can use EIF4G1 as a potential target for therapeutic intervention. So, I mean, to just go back to the history of the world, what we call antibiotics are actually antibacterial agents which only target protein synthesis. Erythromycin, tetracycline, chloramphenicol, and aminoglycosides, they are protein synthesis inhibitors for bacteria. Why? Because they selectively block the 70S bacterial ribosome and do not potentially have a less ability to affect our eukaryotic ribosomes. Then there's cyclohexamide. I was remembering from Bharat's earlier conversation, he said lymphotoxin only was toxic when you were using cyclohexamide to inhibit protein synthesis. But cyclohexamide itself is toxic to the cells. It's highly toxic, and cyclohexamide cannot be used. There is still an unmet need at this point for the agents that can specifically use targeting components which are driving translational reprogramming in cancer. So make this long story short, normal cells have oncogenic addiction and results in a local tumor formation because cells proliferate and they survive. Then they acquire additional changes and become these therapy-resistant tumors. And what is at the base of all this oncogene addiction is oncogenes can drive translational reprogramming, and they depend on this translational reprogramming to do their functions. And how do they do it? This translational reprogramming selectively primes for the proteins. It selectively loads messages that are required for proliferation for the local control of the cell growth, but also selective loading of the messages that are required for cell survival, migration, and invasion are critical for driving the tumor progression from an indolent disease to an aggressive disease. And what we believe is that the EGFR1 is a critical component of this translational reprogramming process. As such, using novel agents, and nothing better than this conference to bring it, the small molecules or nutraceuticals that may target EIF4G1 could have a huge impact on our therapy-resistant tumors in future. With that, I would like to acknowledge my contributors and thank everybody who was part of this process. Thanks so much.
Thank you very much.
Thank you very much, sir. The session is open for questions.
Dr. Singh, can I make a comment and ask a question, please?
Yes, yes, please. Sir.
Yeah. Thank you so much, Dr. Kaul, for showing us this wonderful protein that you talked about. There's a eukaryotic translational initiation factor 4, EIF4G, as you call them. And what is more important, and to our point of view, that this protein is increased significantly in patients of prostate cancer who have a very bad history. And in fact, the survival, if this protein is very high, is minimized. And you also showed us that one can knock down this one or disrupt the EIF4G, the EIF4E complex, and then it could actually be very, very helpful. I was just trying to understand, have you looked at this protein? Of course, you said that you looked into all the available cancer cell lines, but in addition to prostate cancer, any other solid cancer or solid tumor or in leukemias, what is the status of this protein in leukemias?
Thank you for that excellent question. We have only looked in the solid tumors, and almost every solid tumor that we looked at, we find that this protein is highly upregulated, or more than 200-fold levels compared to normal cells. Indeed, normal cells in many cases do not even have EIF4G1, but the tumor cells have high levels of this message, at least, not the protein levels, but the messages we have looked at.
So the prognostic value of this one that we showed in prostate cancer, is it also shown in other solid cancers?
Yes, sir. All solid tumors, the median survival of patients is at least 2 times.
Yeah, so I mean, breast cancer or maybe hepatic or colon cancer.
The data, so on screen, so everybody can see that. This is the data for multiple tumor. Can you put the screen back on? Yes, 1 minute. You can look on the screen, he's going to share it with you. So if you look at this slide, these are the median survival of the patients based on how much of message for EIF4G1 is associated. I think the first one is brain cancer, gliomas, second one is kidney cancer, third one is liver cancer, fourth is lung, mesothelioma, pancreatic cancer, this is prostate, and this sarcoma. There were other tumors that is not listed because the list is long, but these are the data you can clinically look whether they are significant.
Fantastic. Thank you so much. That's really very, very good.
Thank you, sir.
Thank you, sir, for the nice presentation. What happens in the, inhibiting this particular protein, what will happen to the normal cells?
Normal cells, nothing happens because this protein is not even present there. Very low levels are there. The cap-dependent translation is selectively used during the stress phase of the cells or during the repair of the DNA. When they have a DNA damage, they require this process, or when they are in hypoxia. So most of those processes are critical for the tumors only. In terms of normal stem cells, if you look at, we have not looked at it, but there may be something there. But in mice, there was no change in either histology or the weight of the mice during the treatments.
Thank you.
Thank you very much.
Thank you, sir.
Thanks for a wonderful talk. Thank you so much. We learned a lot from this.
Dr. Kaul, very much for a nice talk. In this mRNA, have you looked for the antisense mRNA also for this same protein?
And can you repeat that?
Antisense mRNA.
For what?
For this factor?
No, we have not.
Thank you.
Okay, and there is one more question.
Very illustrious talk, sir. Thank you so much. Yeah, the question is, it was a very nice observation where in the normal cell, this guy is not there. Do you have any idea the knockout, whether it is viable, the animal, the knockout, because certain critical guys, they may not be, but in development they have a lot of roles. So do you have any idea?
There are 2 knockout mice with the 4G1. They are pretty viable and they have smaller body weight, but other than that, they're living.
Oh.
It is not required for development.
That's very interesting. So, a quick question. Is it only one gene for this guy, or there are any?
Only one gene.
Only one gene.
But there are 3 isoforms. I told you. EIF4G has family of 3 members.
Yep.
1, 2, and 3.
Yeah.
1 is selectively upregulated in tumors. 2 is for non-cap dependent translation, is in every cell. And the 3 one does cap dependent translation, is in some cells. So 1 is selective isoform that we target.
Thank you so much.
Thank you very much, Professor Kaul, for an illustrious, very nice lecture.
Thank you.
On metabolic, people were talking of metabolic reprogramming in cancer, and translational reprogramming is coming up as one of the next targeted therapies. It was really inspiring. Thank you very much.
So, I mean, these are not, I mean, many times when we sit in our own labs and in our own rooms, we just think only what we do, but they are all interrelated.
Yes.
Any
exactly.
process in the cell is driven by proteins.
Yes.
Metabolism is driven by enzymes. And if you have enzymes in a different shape and form, we get different things.
And
But
metabolic reprogramming is not possible without
Translational.
translational reprogramming. Thank you very much.
Thank you very much. Dr. Singh, are you continuing, or shall I introduce the next speaker?
You please introduce.
Yeah, the next speaker is online. Professor Dulal Panda is from Naipur, Mohali, and is going to speak about protein aggregation related neurological disorders, natural products as therapeutics. Please show the CV slide of Dr. Panda. Oh, anyway, Professor Panda, there's no CV slide for you. You go on. The stage is yours.
Professor Panda does not need introduction to Indian audience.
Okay, wonderful. That's great. That's great.
Okay, okay. So this is Dulal Panda speaking, and I have been talking in INCD so many years. Today, the talk I'm going to give is probably 100% different than that I used to talk on cancer. So this will be a more of protein regulatory disease. Okay, so there are some diseases which are related to protein aggregation. The most important 2 of these are Alzheimer's disease and Parkinson diseases. So in Alzheimer and Parkinson, there are several school of thoughts. One school of thought is is the aggregation of protein called tau. Another school of thought is the A beta aggregation. But now it has been established beyond a logic that tau aggregation precedes A beta aggregation, and so tau aggregation is predominant in all kind of neurological disorders. So I'm going to the next slide. So in looking at this, that if you look at the axon and dendrites, these are important things that actually carry the signal. I'm not going to discuss too much of this, but there are a protein called tau and MAP2. MAP2 is important for the development of dendrites, and axon, it is tau. And this tau protein actually stabilizes microtubules. It helps to grow microtubules, growth cone development, and it, that actually is important. When it dissociates from microtubules, it aggregates, and the aggregation is important for Alzheimer, Parkinson, all other kind of neuronal diseases. Sorry. Page down. Okay. So the way tau works, actually, this is the microtubule, and tau actually bind along the length of the microtubule, or laterally, or longitudinal manner, or lateral manner. So one tau actually has 4 different repeats. I will show you just a few seconds later that this repeat bind to different subunits of tubulin in microtubules and thereby stabilizes this. Just look at this video. What you are seeing, this is the growth and shortening of microtubules in live. This picture is taken from live breast cancer cells, and microtubules are growing and shortening. And so this is important to capture the chromosomes and align the chromosome on the metaphase plate. So for this is important for cell division. Similarly, for growth development, it is important. So on. And the tau, what that tau does, it actually, yes?
Your slides are not moving. You're stuck on your microtubule slide. We are stuck on slide 4, slide 3. Maybe
yes, I'm on slide 3 only.
We are on slide 2 here.
Okay, this is slide 2. This is slide 3.
It is not here.
The slide is not moving.
Professor Panda, I think you are working with 2 monitors.
Yes, yes.
That is a problem. I think you have to go to display and say replicate the slides on both monitors.
Okay, okay, 1 second.
This is a problem. You are actually advancing from one monitor but not on the other, which we are seeing. Actually, we are not seeing the presentation form. We are seeing the slide forms. Yeah, now it will be better.
Okay, okay. Actually, what happened, I came this just few hours ago, I landed in United States. And from there, I'm just talking. Okay. Initially, I canceled my talk when they said that, oh, it can be done on online. I thought that even I stay and give the talk. So, now, can you see this?
No, it is not. You are actually using the presenter's view. If you see on the monitors, use presenter view. So you just remove the click from there. On the top ribbon, go to monitors.
Okay, top ribbon, go to monitor.
So don't use the presenter view. So we are seeing your presenter view, not the slide.
Achha.
Can you see that?
Now can you see the slide 2?
Yeah, now it's correct. It will be now.
Okay. Okay. So if I go to the next slide. Oh my God. Okay. Can you see microtubules moving? Hello? Can you see microtubule moving?
No, not yet. Yeah, this one is there. Yeah, they're moving.
Okay, so basically, I think this is okay. If I look at this, what you are looking at, microtubule are growing and shortening, and they are very dynamic polymers, and they are important for cell division. Also, they are important for the growth and development of neuron and the growth cone development. And what tau does, that's what I wanted to show you. What tau does, tau stabilizes microtubules, and tau stabilizes microtubules. And see, in the absence of tau, the lower one is growing shortening, growing and shortening. When we add tau, what it does, it persistent growth. Okay, and it reduces the shortening phases, so it decreases the shortening or catastrophe, increasing the rescue, the growth phases. So it helps to grow the growth cone and the signaling and stabilize the microtubules. So that is the function of tau in neuron. This tau now, if you look at the tau structure, it is a very simple protein. It has 4 repeat domains and there are projection domain. And the repeat are very, very similar. That's why they are called repeat domain. And this 18 amino acid repeat, they can actually polymerize microtubule in test tube. And also in cells, it can actually increase the microtubule polymer level. But anyway, there are tau has 2 kinds of tau. One is adult tau, another is the fetal tau. And each adult tau, due to alternative splicing, has 3 variants, and fetal tau has 3 variants. The important thing is the ratio of adult tau and fetal tau actually determines the patient condition and Alzheimer's disease and so on. And just to remember, this simple protein has more than 60,000 papers in PubMed. Okay, and it is really interesting that it is a highly soluble protein, heat stable protein, but this protein aggregates in brain. So what triggers the aggregation, what makes it to get it aggregate, that is important to know. So there are 2 school of thoughts here. One is the tau protein, and abnormal tau due to tau post-translational modification, phosphorylation, mutation, it lead to it loses its ability to stabilize microtubules, and that lead to impair functioning of microtubule, leading to the loss of transport and stability of microtubules that lead to cell death. Another is that this abnormal tau aggregates, and aggregated tau actually kills the neuron. So a normal tau does all the function, but whenever the tau get aggregated or post-translational modification happens, then it actually cannot bind to microtubule, cannot stabilize microtubules, and it also changes the transport. So the cells actually die, or tau aggregation. Today I'm going to talk about this path, aggregation of tau and inhibitors of tau. Okay, so my talk will be solely based on how tau does aggregate and what are the inhibitors of tau aggregations. So there are different tau aggregate inhibitors are known, and some of them actually have moved these. These are some of these from our lab. Okay, so I'm going to talk about a compound called Sikoni, and this is a natural product isolated from a plant, and this is used in Chinese medicine, and it has been shown to have neuroprotective activity both in neuronal culture level as well as in mice level. So we started looking at this, whether it can inhibit tau aggregation and what kind of effect it produces in cells. So this is the tau construct, and there is no tryptophan. We actually introduce one tryptophan in R2 repeat, and that gives us a handle to monitor the tau aggregation. So here, when tau aggregates, there is increase in tryptophan fluorescence, and so if it does not inhibit tau, if it does inhibit tau aggregation, so there will be a decrease in tryptophan fluorescence. So we actually got a lot of kinetic parameter done to see how does it inhibit tau aggregation, and from it inhibits the rate of tau aggregation. It also delays the tau aggregation, and so on. So that indicating that it can actually inhibit aggregation of tau. Then we used actually heparin-induced aggregation of tau for this, and we are looking with a marker that is thioflavin-S. When tau aggregates or anything from amyloid, thioflavin-S bind and the fluorescence increases. So the increase in fluorescence is a marker of aggregation, and we are using chiconin to see whether it can inhibit. So what we found that it inhibits the tau aggregation using thioflavin-S fluorescence, both for a 6 hour and 12 hours. The difference in time is that the longer we incubate, the more bigger filaments are formed. Initially, it forms the oligomers, and with time that it forms the filaments. I will just show you the data. We also use different type of aggregation inducers, for example, RNA, academic acid, because none of these are, except RNA, academic acid or heparin, these are not available in brain or in a very low concentration. So in vitro, we use all sorts of inducer to induce the aggregation of tau and see the effect of chiconin. And in all cases, it actually inhibits tau aggregation. And if you look at the data with the inhibition, using the AFM, and you see the bigger oligomers, and with the chiconin, there is no aggregates. So that means it is able to actually inhibit the oligomerization, and the oligomers are actually highly toxic. I will show you a little later. Using dynamic light scattering, there is which can measure the particle size. A lot of nanotechnology people use this, and there is actually decrease in tau oligomerization. And also we use dot plot assay to show that there is it actually inhibit aggregation and SDS page. So many different assays we have taken to show that it not only inhibit the rate, but it also inhibits the tau oligomerization. As you see, this is the dynamic light scattering and a dot plot assay, and looking at the SDS page. Then if we incubate tau little longer, it forms filaments like this. Okay, and these filaments are very stable filaments. So in presence of chiconin, actually, there are only few filaments are seen, both by electron microscopy as well as the atomic force microscopy. There are only few filaments are seen. So that means that it is able to inhibit the transition from oligomer to filaments also. Now there is another technique that is coming for liquid phase separation, and you can actually see these liquid droplets. Many biological processes are now getting explained using the liquid droplet formation. Here what we did, we make the droplet, so tau actually form the droplet in presence of inducer. And then you can look at the droplet. This is a small droplet and the magnified view of the droplet. But in presence of chiconin, there is no, actually, there are only few droplets. They are smaller size and number reduced. That shows that it cannot form the tau oligomers. And this is this assay is really an interesting assay to show the droplet formation. Okay. Now we have standardized another method where actually we label tau with FITC, and that tau in presence of an inducer, it forms a droplet, and we can look at the droplet. And what we are doing here, using the FRAP, fluorescence recovery after photo bleaching, we actually bleach that and allow this fluorescence to grow. And this is the control tau, and this is in presence of chiconin. That means chiconin actually increase the dynamics of tau droplet formation. That means that tau droplets are not so stable in presence of chiconin. And the half-life is half-life in presence of chiconin is say 0.069, whereas in presence in the absence of that it is 44 seconds is the half-life in the absence of chiconin, and 11 seconds is the presence of chiconin. So nearly 4 times reduction in half life, indicating that tau droplets are highly dynamic in presence of sequonin. So sequonin also disintegrate the tau filaments, and both by DLS as well as this thioplombin explorations as well as electron microscopy, we have shown that actually if you make tau filaments and add sequonin, it actually disintegrate the tau. See here there's some AFM microscopy images where the tau filaments are seen. When we add sequonin, actually dissolve the tau filaments. So that is amazing that it has the ability to actually disintegrate tau filaments. Now, in order to look at the binding of sequonin to tau, we use some biophysical technique to show that it actually binds to tau and induce conformational changes in tau. Then we wanted to look at whether it has the ability to inhibits tau-induced cell death. So we actually make tau oligomers and add the oligomers in cells, and the tau oligomers by get into the cells and kill the cells. But in the presence of sequonin, the cell death is minimal. So that means it can protect the tau oligomer-induced cell death. So that is remarkable. Now we needed to see how we can do these experiments in animal to see whether whatever in purified protein and culture level we got can be reproduced in animals. Then we have a potential molecule for neurological disorders such as Alzheimer or Parkinson. So this is this work is published, and what we are looking at different assays to look at the tau oligomerization and inhibition of tau oligomer formation using sequonin. A few years earlier, we also started looking at ability of curcumin to bind to tau, and can tau actually differentiate between curcumin can differentiate between 4R tau, that is adult tau, and 3R tau. Remarkably, curcumin has a much stronger binding with the adult tau compared to the fetal tau, and the effects of curcumin is much stronger on the adult tau. I'm not going to go all the data, but just to show you that it binds to the microtubule binding domain of tau and inhibits actually tau aggregation. And since I think I'm going closer to time, so there are a lot of interesting data where we had showed that curcumin can inhibit tau aggregation. Also, filaments, it can disintegrate tau filaments, it can dissolve tau filaments. So some level of curcumin might be helpful on doing this. So so there are a lot of data. I'm not going to show this, and it's getting very midnight for me here. So lot of students actually done this work in last 5 years, and I thank them for the work, and I actually thank the organizer for inviting me and to give this talk. Thank you, and I would be happy to take the questions.
Thank you, Professor Panda, for this wonderful talk. The session is open for any questions. Yes.
Thank you, Professor Panda. I am Professor Chandishi Mandal from Central University of Rajasthan, Jaipur. It's a very nice talk, and it's a kind of new information about the tau aggregation, and it's related to Parkinson and Alzheimer disease. And you have mentioned that tau can destabilize the microtubule formation, and when the tau becomes aggregated, at that time the tau can not do this thing, right? And the second one, your sequonin, it inhibits the oligomerization of tau. What happened about the impact of sequonin on A beta, whether it can also inhibit the oligomerization of A beta? This is 2 questions from my side. Thank you.
You see, when tau get post-translational modification, for example, acetylation and any other phosphorylation, then it cannot bind to microtubule. So that is one hypothesis. Another thing is that when tau gets post-translational modified, they get aggregation also. That's another. So which one is predominant? Still we don't know. When we add sequonin, actually we found out sequonin inhibits tau, all step of tau oligomerization, from LLPS, liquid-liquid phase separation, to tau oligomer formation, and to tau oligomer to filament formation. In all steps, actually it inhibit tau aggregation. So we have kinetic data and the microscopic data that I showed you, that in all steps of aggregation, from nucleation to elongation, and then phase transfer, as well as the filament formation, even it can actually dissolve the filaments. These are all kinetic parameters, all interlinked parameters. Sometimes some molecule may not have that ability, but this sequonin does it. So in all steps, so far what we have seen. Okay.
Thank you very much, Professor Panda. I think that's all. Thanks.
Dr. Singh, there is a question in the chat box by Dr. Namita.
Okay, there is a checkbox question. Fine.
Yeah, she wants to know, is it clear about tau protein aggregation? Which form gets aggregated?
See, that question is, I can answer it, that tau 3R get aggregated, tau 4R get aggregated. All forms of tau are known to aggregate. But the whole question that is not settled, even after 60,000 papers in PubMed, if you just put PubMed tau, you'll get that. Why does tau aggregate? This is a highly soluble protein. You can actually, the way people isolate this protein, and see, if we isolate from brain extract, okay, if we take any animal brain and boil it, almost all protein will precipitate. The soluble protein will be tau. And nowadays we isolate it is expressed in bacteria, and then lyse it and then boil it, and almost all protein get precipitated. Then we do the chromatography, but the tau protein remain. So still people do not know why does tau aggregate. And that's why people started looking at this LLPS, liquid-liquid phase separation, and all these things. And maybe these new techniques are giving some ideas what triggers the tau aggregation. Okay.
In continuation of that, could I ask, is there any human data, either in Alzheimer's or in Parkinson's, about the aggregation of tau?
Yes, yes. There are tons of data from patient showing the tau aggregates, and the tau aggregates are so stable that it requires, HCl2 actually. If you take the sample from diseased brain, okay, human brain, and you dissolve in acid, even still remaining part, what is 33% of the protein get dissolved, and these are the tau filaments. So it is like rock. You cannot actually take a hammer and break it.
Okay, I think Namita has raised her hand. I'm sure we have already asked the question that she wanted to ask. Thank you very much.
Thank you.
Thank you. And now, Professor Mehra, you can introduce the next.
Okay, the next two talks are actually short lectures. And the short lecture one is by Dr. Lakshmi Mundakur. She's from Sami Sabinsa Group Limited, Bengaluru, and going to talk about herbal supplements for non-alcoholic steatohepatitis, efficacy, mechanism, and clinical evidence. Dr. Lakshmi, please.
Just a minute now. Just.
Safety, please stand clear of the doors. We thank you for choosing SpiceJet.
Our next speaker is Dr. Lakshmi Mundukur.
Yes, you can.
Thank you, sir, for the introduction. I'm here. I think the slides are taking a little time.
Okay. Yeah, there you are on the screen.
For giving me this opportunity, and it is a great privilege to talk in the same session as Dr. Talwar and Dr. Kaul, who are pioneers or who are stalwarts in this area. So I come from an industry, so my talk is not going to be on basic science, but I'll try to cover what we have done in the last few years. So I'm taking you away from cancer and going into an area of metabolic diseases. Yeah.
Cancer is also a metabolic.
Yeah, sure, absolutely. But then I'm so a little introduction about our organization. This SAMI Sabinsa group was it was established in the year 1988 by Dr. Majid, and he reverse merged it. In fact, it started in New Jersey, USA, and like Indian operations started in 1991. And then in 2003, it was reverse merged, and the SAMI group in India became the main component, and the Sabinsa became its subsidiaries. And the Sabinsa we have in all over the globe. And the main, the important about our industry is we look from optimized cultivation, and we took a lot of efforts into conservation efforts, and we know our responsibility. So we work with local banks, insurance groups, with the local farmers to get our products. So we are quality driven, and we are ISO 17025 accredited, and our manufacturing plants are CGMP compliant. So this month of October is declared as the National Liver Awareness Month. So I thought, why not talk about liver diseases in this particular conference? So coming to the liver, it's the only organ that can completely regenerate. So I'm not going into all these details because all of you are much more aware about the liver probably. Then these are some of the common are the fatty liver. Accumulation of liver, and this perhaps isn't to some extent, may not be to a great level, but some extent of fat accumulation is there in every normal individual. Then hepatitis, then cirrhosis, and liver cancer, of course, is these are the common diseases of the liver. Now, the markers are, of course, the liver enzymes, bilirubin, alkaline phosphatase, and then liver disease. What is it? So, as the name suggests, non-alcoholic. So, it's not driven by alcohol. So, in the absence of alcohol, when fat builds up in the liver, so that is characterized as non-alcoholic fatty liver disease. Now, this is in the early stages of the disease, it is just the fat accumulation, which probably is not too much of an issue. But if it starts progressing into an inflammatory stage, then it is known as steatohepatitis, which is known as NASH. And then the chronic condition, it can go further into cirrhosis and liver cancer also, which becomes very difficult to cure, and transplantation becomes the only option. Now I'm trying to tell you what are the risk factors for NASH. Obesity, of course, is one of the major factors. Overweight, like when you put on weight and fat starts getting accumulated into your liver. Now, type 2 diabetes, insulin resistance, PCOS, underactive thyroid, all these could be some of the cause factors for developing NAFLD. Now, as I told you, it could be a simple fatty liver to start with, then goes on to develop NASH, fibrosis, and then the cirrhosis of liver. So if you look at the progression, NAFLD is present in almost 15 to 25% of the people. And out of that, around 40% develop NASH. And then out of that, 20% go on to develop the fibrosis and then the cirrhosis, and then it is the hepatocellular cancer. Now, if you look at the prevalence, now it was around 25% maybe 6-7 years back, but now it has reached to a stage where in USA, 47, almost close to 50% of the people have NAFLD. So it's alarming increase in the disease, which I think we should look into, because it's a silent disease. There is no manifestation which is happening. So you probably don't even realize that I have the disease unless you go to the later stages. So what is the pathology? The pathology is lipid accumulation. That is mainly because of fatty acid uptake, and VLDL export is inhibited. So, and then de novo lipogenesis and fatty oxidations are inhibited. Then comes the inflammation part, the macrophages which get activated, and then they start secreting some of the inflammatory markers which activate the hepatic stellate cells. And hepatic stellate cells are the ones which will this is an EMT transformation, like a lot of people have talked about it earlier. So the TGF SMAD signalings promote the collagen synthesis and ECM deposit. So that is what leads to the fibrotic stage. Now this is just to tell you what happens in this. So I'll skip this. And then coming to the treatment, very surprisingly, though it is a disease which is affecting 50% of the population, there are no drugs available in the market. Now, lifestyle changes, exercises like caloric restriction, and only vitamin E has been used to some extent, and diabetic medications have been used. Metformin is known to have some effect on NASH. Now, the drugs which are in the pipeline are targeting lipid metabolism pathways, and obeticholic acid is one promising drug which is in the phase 3 trial. Insulin sensitizers are being used, and GLP peptide receptor agonists are also being used for the treatment, but all these are in the experimental stage. Nothing has been approved by FDA. Now, coming to alternate therapy, yes, there are plenty of phytochemicals which have been tried in the preclinical stage. There are not many clinical studies, but preclinical, a lot of these polyphenols have been used. Some of them being still beans like resveratrol, the flavonoids, the anthocyanins and quercetin and all that, phenolic acids, lignans and curcuminoids, of course, have been experimentally proven to help in NASH. Now, we wanted to look at this with a combination therapy. Now, curcuminoids is already known. It is known to help in regulating the fat metabolism. And then we have had some data on this particular plant, Garcinia indica. It is nothing but kokum, which is available in the coastal regions of our country. So kokum is a fruit, and the juice of it is being taken in Goa and Mangalore as mainly in summer to reduce your heat. So we isolated one particular compound from this Garcinia indica, which is known as garcinol. Garcinol is so I'll come to it. So curcumin, all of you know, it's one of the well-studied molecule. So what we have is a 95% curcuminoids. So coming to curcuminoids, it's not a single molecule. So though we talk about curcuminoids as a single molecule, it has 3 different polyphenols. It is curcumin, dimethoxycurcumin, and bis-dimethoxycurcumin in a particular ratio. And that is what is the curcumin that we normally talk about. So this plus the garcinol that I isolated. And the garcinol is standardized to a 20% final, because we purify it to 80% and then bring it back to 20% because that is the one which is stable. And we use bioperine or piperine at a very low concentration as a bioavailability enhancer. So now we did some in vitro studies, in animal studies, and then the clinical studies. So I'll take you through. I won't go into the details of anything. Try to finish my talk in 20 minutes. So we did this I'm not talking about the in vitro studies. I'll directly go on to the in vivo studies. This was NASH fibrosis model, which we did along with SMC labs in Japan. Now, this is a model which closely resembles what happens in the human condition. Now, in this, the 2-day-old mice is given the first hit, which is the streptozotocin to induce beta cell injury. And then from 5 to 6 weeks onwards, the mice are kept in high-fat diet. Now, if you look at, there are different stages of this. So up to 5 to 6 weeks, you start steatosis appearing. Then it is the steatohepatitis state, and then the fibrosis, and then so it follows the disease pattern which is seen in human. So we stopped our assay after the 9th week because we didn't want to go to the later stages. We were looking at the early stages of the disease. So this was the protocol we followed. So just to tell you that we used curcumin C3 complex alone, and we used the garcinol alone, and then we combined garcinol and curcumin. So if you look at this, the first is the NAFLD activity score, which is based on the histological sections which are stained with hematoxylin and eosin. So if you look at the data, the combination was significant compared to the both curcumin and garcinol alone. So, and then the steatosis score also came down, and also the ballooning. Ballooning is something which happens in the liver. Like maybe the next slide will show you the ballooning much clearly. If you look at the first one, you can see that the cells have bulged. So this is known as the bulging of hepatocytes, and the bulging came down when we treated them with the combination. And also this is stained with the collagen staining, which is done by picrocereus red. You can see all those red stains are the picrocereus one. So the collagen level also came down. Now then the collagen, the precursor for collagen is liver hydroxyproline, which was estimated, and curcumin and the combination were able to reduce liver hydroxyproline. And when we looked at the gene expression based on the fold changes, the TGF-beta and collagen 1 levels were lower. Now, then we looked at the TNF-alpha in the liver tissue, where we had lower level of TGF-alpha both in curcumin and in the combination, and that was probably mediated by the NF-kappa B phosphorylation. Now, then we went on to look at some of the biomarkers. One of the main one was the adiponectin and the FGF resistance, which develops in NAFLD patients. So we had the curcumin was not very effective in adiponectin, but garcinol was little better than the combination actually. And then, but FGF resistance was brought down by the combination. Then, of course, since both of them are antioxidants, there was a reduction in most of the markers of antioxidants. So, in conclusion, we can say that garcinol is able to reduce hydroxyproline, decrease in the trend for TGF-beta, collagen, fibrosis. The combination has significant decrease in both fibrosis as well as the NAFLD activity score. So we published this data, all the in vivo data, and then we went on to do the clinical validation. So the garcinol, curcuminoids, and piperine. So we call this GP, or our trademark for this is live longer. Now, clinical study was done. It's a preliminary clinical study, which was done as a two-arm randomized double-blind placebo-controlled study. And garcinol was at 50 milligram and curcuminoids were 250 with 5 milligram of piperine or piperine as bioavailability enhancer. These were male and female patients up 30 to 63 years who were diagnosed for NASH based on elevated liver enzymes and ultrasonography. Then they were obviously having a higher BMI, and then GCP was given twice daily. for 90 days. And apart from the intervention, we also ask the participants to refrain from taking any processed food and not have junk food and have 30 minutes of exercise every day. So the primary objective was to look at mean change in liver enzymes and then the liver fat content, which we measured by using a fibroscan. Fibroscan is one of the recent development in the diagnosis of NASH. So which measures both the steatosis or the accumulation of fat as well as your stiffness. The stiffness talks about the fibrosis in the liver. So we looked at that as one of the markers. Now, secondary objectives were the liver lipid profile, the glycemic profile, the biomarkers, and then changing BMI, the weight, and then the, of course, the safety was one of the major concern, which was the secondary endpoint. So I'm not showing you all the data because this our clinical study is yet to be published. So I'll show you some of the important data, which is the reduction in AST and ALT in GCP or the treated patients compared to the placebo. Then the liver stiffness and fat deposition. So this is a non-invasive technique. So the FAST score, which is becoming very important in looking at the liver stiffness and steatosis, is this score is developed by using the fibroscan data.
Please
on your steatosis.
Wind up because time is over.
This is one of the last slides. I'm not taking too much time. So the body weight, there was a reduction. The waist circumference there is a reduction, and adiponectin was increased, and while IL-6 showed a good reduction here. So again, when we come to safety, GCP was it did not cause any changes in the serum, any of the hematology or other markers, and it is a safe nutrient for consumption. So we have also done the preclinical safety for this molecule. So all the GLP studies have been done to show that it is safe for human consumption. So I think I should thank you for your patient listening. And if there are any questions, you can.
Thank you very much for the talk. And
I have one question.
Yeah, sure.
So in your GCP trial,
yes, sir.
What are the inclusion and exclusion criteria?
So I think it was there in one of the slides. I didn't go into the details. Yeah, the main inclusion criteria was they needed to have a NASH which was diagnosed by the liver enzymes and the ultrasonography, and the stage of liver steatosis was less than, the grade was 2. It was, we didn't take something mild to moderate NASH. We did not take the severe cases.
And mode of delivery? Orally?
Yes,
is oral delivery? Or?
It is oral delivery. It's an oral.
Thank you. Thank you, Dr. Singh. I agree with you. Because of lack of time, we cannot go on and on. I just wanted to make just one remark, and that is that NAFLD is now being called as MAFLD. It is a metabolic associated fatty liver disease. But anyway, there are quite a few things that I had in my mind to ask you, but because of lack of time, we'll talk some other time. Thank you so much for your.
Agree. It is now known as the metabolic disease of the liver.
Yes. Yes.
Okay. Thank you very much.
Thank you so much for your time.
Thank you.
Okay, so our last speaker in this session has 10 minutes to talk about. This is Mr. Takeshi Kobayashi from Tokyo. He's in the Fujifilm company. Fujifilm Wake Pure Chemical Corporation product related to pharmaceuticals and nutraceuticals for cancer and other chronic diseases. So you're going to give us a sort of view of what kind of agents you have in your company. Thank you so much. Please go ahead.
So I appreciate for having the good opportunity for introduce about our company. I am Takeshi Kobayashi of the international manager of the international sales section from Tokyo, Japan. our company was founded in 1922 in order to develop and manufacturing deliver the research purpose reagent for the market. Anyway, so I wanna say, so our company has a very long time history, but yes, then another 5 years ago, our mother company Fujifilm merged by merged for our company Waco in order to develop our business to the global market. So we used to focus on the only domestic market to Japan, but from 3 years ago, we tried to expand the business for a global market in order to contribute to the scientists in India together with our business partner, Amit Lab Service. Our company's main product is mostly so life science related research purpose products and the chemical products such as a solvent. We have around 76,000 kind of products. It looks like a smaller version of the milk and Sigma Aldrich. In domestic market, Japan is the top share for the reagent market and the life science and the chemical related research purpose products. This is some our main products. For example, neuroscience field, the anti-IBA1 antibody, it's a microglial marker, and amyloid beta antibody, and then exosome isolation kit, and Lister end peptidase, which is a cleavage enzyme. These products would be the top share in the global market. Anyway, so I wanna say our main mission is to contribute to the global scientists. If you wanna looking for the some research purpose products, could you please visit our website, Fujifilm Waco Pure Chemical. Okay, Dr. Nidi, could you take over my presentation?
Hello.
Hi.
Hello.
Hello, yes.
So,
thank you very much, Takeshi-san, for giving the presentation, for briefing our product range. Now, for the related to the cancer sciences and all, Mr. Shubhankar will brief some of the products which we have in Waco. Thank you very much, Shubhankar.
how long will that take actually?
Only 5 minutes.
2 to 5 minutes.
Now, why do you keep changing from one speaker to the other and then to the third speaker? Yes, please. I hope the third speaker is the last speaker.
Yes,
exactly.
In the bucket of Fujifilm Waco, we do have some ELISA kits for the detection of exosomes and extracellular vesicles. As you already know, exosomes are the vesicles generated by all cells that can carry nucleic acids, proteins, and metabolites. And for detection of that, these ELISA kits can be used. We have different types of ELISA kits based on protein serine method. We'll show you that some of the details for the ELISA kits. These are some PS capture ELISA kits which can capture the exosomes and they can show the result as per their sensitivity, and their features are 50 to 100,000 folds more sensitive than Western blotting. In the Western blotting, they are directly relative to the quantitatives of the extra vesicles, and they are available in various detection antibodies like CD8, CD61, and CD83. They need only 1 to 1 of the 10th of the small samples amount of the Western blotting as per the human serum and plasma. These are some details for the serum as per the sample for the serum, as well as the cell culture, super-rated cell cultures. We do have some data as per the cell cultures of COLO201 and IPC cells, which can be, you can say, the extracellular stem cells. which will show you the absorbance as per the OD and all the things. These are some specific related to the antibodies like CD9, CD63, and CD81, which can capture the antibodies of the human, which is based on the streptavidin hub protein, which is a conjugated of the E. coli, which is a conjugated protein extracted from the E. coli. This is the principle based on biotin method, how ELISA kits detect the hub protein and bind to that. After that, they can show the growth of the cells in the ELISA kits, specifically for the CD9, CD63, and CD81. These are some dilution samples for the human serum for the different antibodies. These are the results for this culture supernatant sample for the COLO201 cell lines. Thank you.
Okay, thank you very much. And with this we come
to come to our booth whenever you have time. Thank you very much. Thank you for the organizers. Thank you very much.
We come.
I think with this, we come to the end of this session. Sorry for being a little late. We have held you from your lunch. So now we all break for lunch, or we pass it on to the organizers. I want to thank
Professor Mehra,
Professor S.M. Singh.
At lunchtime.
Yeah, I'm also going to take my lunch. Yes. Dr. Singh, thank you so much for your cooperation, and really you conducted it very well right over there.
Thank you very much, Professor Mehra and Professor Singh.
Mic is on, of course.
We now conclude the session, and I would request Professor Singh to present mementos to our speakers of this session. Professor Talwar. Next, we have Dr. Lakshmi Munkar. Next, we have Dr. Lakshmi Munkar, please. And next we have Mr. Takeshi Kobayashi, if someone can receive the memento on his behalf. And now I would request Professor Bharti to present the memento to our chairperson, Professor Singh. Also, we had missed out on our earlier speaker, Dr. Rekha Kumari, so I would request Professor Singh to present a memento to Dr. Rekha. Professor N.K. Mehra's memento, we would arrange for shipping.
Please give it to Dr. Alok Bharti. I like his color of his shirt.
Will do, sir. Will do. Thank you very much, everyone, for being here. We break for lunch now, and we would be meeting for the next session post-lunch. Thank you. So post-lunch session begins at 2 o'clock.
लाइव स्ट्रीम बंद करो जब तक दूसरा सेशन ना शुरू हो जाए। HDMI यहाँ पर कब भाई HDMI गया थैंक यू हाँ ये 4A आ गया ये होने वाला है ना अभी ठीक है और अब इससे होगा सर अब बस इससे करेंगे सर ठीक है
%97. Oh, Anshul Bhaiya.
ऑनलाइन
cancer. So chairing the session, I am pleased to invite Professor Gopal C. Kundu. He is the Director R&D at Kalinga Institute of Industrial Technology. His area of interest is tumor biology, cancer stem cells, and tumor-stroma interaction, among many others. Professor Kundu has done his PhD in chemistry from Bose Institute, Kolkata. He has several postdoctoral fellowships awarded to him from the United States. He was the Dean and Director at NCCS Pune, India. He was the Professor of Medicine and Biochemistry at School of Biotechnology. He has over 116 scientific publications, 1 US patent and 4 Indian patents. He has received a fellowship award for research excellence from NIH USA, and he is the recipient of the prestigious Shanti Swarup Bhatnagar Award from CSIR. The other chairperson, Professor Ajay Kumar, will be joining us in a little while. I would like to request Professor Kundu to please start the session. Okay. So we also have the second chair, Professor Ajay. He has done his PhD in biochemistry from Amala Cancer Institute, University of Calicut. He has developed several novel drug targets for oral cancer and several formulations of drugs for better drug delivery and bioavailability. And he has discovered over 40 potential anti-cancerous drugs. He has more than 38,000 citations and 282 publications to his credit. I welcome you, sir, and we will please begin with the session. Thank you.
So good afternoon. And our first speaker will be Dr. Ruby Anto. She is scientist G, Rajiv Gandhi, RGCB Trivandrum, Rajiv Gandhi Center for Biotechnology. And her area of interest, bioprospecting of natural products having chemotherapeutics, chemopreventive, and chemosensitizing potential. And Dr. Ruby will be talking on pharmacological targeting of MEK-ERK by tryptanthin attenuous melanoma invasion and metastasis through suppression of MITFM. Dr. Ruby also received multiple awards, including National Level Women Bioscientist Award, Fellow of National Academy of Science, Fellow of Academy of Science Trivandrum, and Global Health Award, and published large number of papers in peer-reviewed international journal. Dr. Ruby, please.
Thank you for the nice introduction. Let me first thank the organizers, especially Alok and Anju, for giving me to present my work here. And my lab, as Dr. Kundu said, is focusing on identifying anti-cancer molecules. And we have identified some compounds having anti-cancer potential, chemosensitizing efficacy, and as chemopreventives. And today I will present the work which we have done on tryptanthin, a compound we isolated in our lab. The compound was found to be very effective against skin cancer. So a part of the work I will present here. So yeah, the compound was isolated from Wrightia tinctoria, and the name of the compound was tryptanthin. And tryptanthin has been reported to be very effective against other cancers also, breast cancer, leukemic cells, etc. So we conducted a screening in different cancer cells of different origin, and we found that the compound is most effective against skin cancer. This is non-melanoma skin cancer. This is melanoma skin cancer. So we conducted an evaluation of this compound in both the cancer types. So today I will present the work we have done on melanoma skin cancer. So second. Yeah, since it was found to be very effective against skin cancer, melanoma, we evaluated its efficacy in different melanoma skin cancers and in normal melanocytes. The compound was safe up to 20 micromolar in normal melanocyte, whether it was cytotoxic to all the melanoma cells. And it induces ROS mediated activation of apoptosis in A375. Actually, this A375 cell, it was a melanoma cell. This was found to be most sensitive to this tryptandrin. So we selected this cell line for our further studies. So it induced ROS mediated apoptosis in A375 cells and different, apoptotic related molecule, all the molecules were, the P53, BAX, TBID, all this, and there was a downregulation of BCL2 and BCLXL, etc. So it was a, the mode of apoptosis, the cell death was apoptosis. So we conducted an animal experiment. The corresponding concentration up to this 12 milligram per kg was corresponding to the 3 micromolar IC50. So we transfected the cells with the Luke and we injected orthotopically to the NOD skid mice, and we could find, we can find a very good tumor reduction. This is the control animals. This is the treatment animals. The fluorescent, the luminescence also was very less. So even though this was not a metastasis model, we could find that this orthotopic tumors were, the metastasis into different organs of the mice, and that metastasis was very less in the case of the tryptandrin treated group. So these were the, a comparative evaluation of different organs. And the tissues we checked for, the expression status of the, we can see the PCNA, the proliferative nuclear antigen was completely downregulated by tryptandrin, and the VEGFR2, then BRAF. BRAF is a very important molecule in melanoma, and it was significantly downregulated by tryptandrin. So since we found that.
Dr. Ruby, I think your slide's not moving.
Why?
Whatever you are talking, it's not correlating with your slides.
So, I think the net is not working, right?
Yeah, internet.
Internet is not working.
Ma'am, you're on 10th slide.
You want a laptop?
No,
which has loaded this thing? Then
from here you do it like this you have to do.
From?
Ma'am, you have to click it here and it will be visible. Okay.
I cannot use this thing.
Mouse.
Mouse?
1 second.
I don't know to do this. It's very difficult for me. If I can use a mouse, it is okay. Can I use my laptop?
Ma'am, wait a minute. Yeah, it's working now. Ma'am, please click it here from here only. Like
From
this.
here? Like this?
Yeah, you can do.
Here?
Let me show you. Click tab here.
Yeah.
Like this.
Okay.
Like this.
Yeah. So it has reached here only, right? Yeah. So the compound is inducing apoptotic mode of cell death, and yeah. Okay. So the tumor reduction study was conducted using the A37 cells transfected with the Luke. So we can see a significant tumor reduction with the concentration equivalent to the 3 micromolar concentration, that is 12 milligram per kg body weight. So even though this was not a metastasis model, we could find that these cells were, the melanoma cells were metastasized to different organs of the tumor bearing mice, I mean the control mice, and this was very less in the case of triptandrin treated mice. No? Yeah. So the different molecules related to melanoma, this PCNA is a proliferative nuclear antigen. It is a common cancer marker, and this BRAF
Can
is
you
a
move
very
it in
important.
presentation mode?
Okay. It's not coming?
Yeah,
Can
now
you-
it's fine.
No, he is telling. Is that okay?
Yeah, now it's fine.
It is very slow. It's, yeah, after clicking here, yeah, that's right, yeah. Yeah. BRAF is a very important molecule in melanoma. We can see that it is significantly downregulated by this molecule. So, but when I go to this one, how can I click? If I have to click on the left side, I have to see that, right?
Like this.
Huh? That's what I was doing. Now it's working. Okay. Yeah. So this is a metastasis model. We injected the mice through the tail vein. So these are, we did 2 metastasis models. This one is using, this is a mouse melanoma cell line B16F10. Here we inject the mice through the tail vein. It is black cyst mice we are doing. So the melanoma cells will go to the lungs of the mice. This is the control, and this is the treated group. Okay, this is the NOD skid mice. Here also we are injecting through tail vein. We can see this is the control, this is the treatment group. So these are the tumor nodules produced in the lungs of the mice. These are the treatment group. Here also we can see some tumors, but it is very less. So triptandrin prevents the melanoma metastasis. Sure.
Slide number.
This one.
This one. Yeah, this one.
Yeah. Okay.
Slide.
I think I have to do like this because we have to only when we click on the slide, it will come. Okay. Yeah. So we found that the compound, in the tissue level, we found that there is a BRAF inhibition, and most of the melanoma drugs are focusing on, are targeting BRAF. Okay. So since we found a very good inhibition of BRAF by the molecule, we thought that maybe triptandrin also working through this way. Okay. So, and we checked, we could see that BRAF is completely inhibited by, these are different melanoma cells, and the BRAF is downregulated by the by triptandrin in all these cells, and there was a very good glide score also for BRAF, and it was noted that BRAF is in, triptandrin inhibits BRAF, in mutated BRAF, not the wild type BRAF. Okay, so we thought that definitely the mechanism of action of triptandrin also may be through inhibition of BRAF. Yeah, but we can see here, Here, this is a mutated BRAF, and this is a wild type BRAF, and this is also mutated BRAF. You can see the difference in the IC50. A375 IC50 is 3 micromolar, while the others are 15 micromolar. So there is something else. Definitely triptandrin is inhibiting BRAF, but there is some other molecule which is regulating the anti-melanoma activity of triptandrin. Okay. So we did a microarray. The transcriptomic analysis revealed, it identified 4 sets of molecules. Yeah, this one. I am just clicking on here only.
Wait a minute. Systems.
Yes, can you connect it with my computer, laptop?
Yes, working.
Huh?
Working. Little networks.
Okay, so we could see that for signaling pathways, MAP kinase pathway, AKT pathway, Wnt signaling, and MITF signaling. And we found that all these 4 pathways are converging in this molecule MITFM. Okay, so the MITFM, that is the microphthalmia associated transcription factor, is the master regulator of melanomagenesis. So we got a indication that maybe MITF is the regulator in this triptandrin induced melanoma activity. So we checked the melanoma. Oh. Yeah, yes. The expression of MITFM, it's gone. I think, no, this one. Yes. We can see these are the different melanoma cells. Here we can see the MITF expression status is A375 is basal. Okay, so in all the other cells, the MITF is overexpressed. So maybe that is why it is the IC50 of triptandrin is 3 micromolar in A375 and very high in all of the other melanoma cells. Here also we can see the BRAF status. So here it is mutated BRAF, this is mutated BRAF, this is wild type BRAF. So the cytotoxicity of triptandrin is not dependent on the BRAF status, even though triptandrin downregulates BRAF. But here it is highly correlating. Here it is basal, here on all the 3 it is overexpressed. No? Okay, here, double, huh? There. It's come?
Yes, working.
Yeah, it is not the question of operating. It is time. You need time. Yeah, I am doing that, but he is standing here. But when okay, it needs time. That's all. Yeah. So we can see in different constant the activity of this. This is B16F10, triptandrin dose dependently downregulates, SKMEL28 downregulates. It and the IC50 concentration. So in each cells, triptandrin downregulates completely the MITFM at its corresponding IC50 concentration. Yeah. Then to confirm this, we overexpressed MITFM in A375 cells. It was 3 micromolar. When we overexpressed MITFM, you can see it is from 3, it went to 15 micromolar. Okay, the IC50 changes from 3 to 15 micromolar, confirming that the regulator is MIT-FM. Okay, so yeah, this is the A375 neo cell, this is A375 MIT-F. Yes. Then we conducted an animal experiment using, in the A375 loop. This is only neo cells. Here it is A375 MIT-F. Even at 45 milligram per kg, that means it is 4 times the concentration of the IC50 dose. There was no tumor reduction. But at 60, it is corresponding to the 15 micromolar concentration, there was complete tumor reduction. Okay, that's the same animals, we can see the difference. Okay, 12. Here, this is the transfected cells that only at 60, that is corresponding to the IC50 dose, there is a. But we found that at this 48 milligram per kg, the tissue, the BRAF is completely downregulated. There was no BRAF at all. But still, there was tumor. Okay. Yeah, this is the solid tumor model. We can see the different organs, fully metastasized, while in 60 there was no metastasis. Yeah, this was another experiment. That means instead of A375 meters transfection, this SKMEL28 has the same endogenous expression of MIT-F. Okay, here also the IC50 is 15. So in this cells also, when we injected at 60 only, we got the tumor reduction. Again, confirming the role of MIT-FM. This is the exact in SKM. So this is just a comparison. Here we can, at IC50 concentration, there is no metastasis. Here, only here, this is the IC50 dose, no metastasis. And this is SKMEL28, this is IC50 concentration, no metastasis. So here is the comparative evaluation of a MIT-F expression status and BRAF status. We can see that the compound completely downregulate both BRAF and MIT-F. Okay, and as I told you, here we at 60 only we could show a complete downregulation of MIT-FM, but the data is not put here. At 48 itself, we could find complete downregulation of BRAF. So there was several reports that MIT-F is responsible for the chemoresistance of imirafinib and dacarbazine. Both are very important drugs for used for melanoma. And triptandrin also downregulates the expression of BCL2. This is a major downstream target of MIT-FM. Here also we got a complete downregulation of BCL2.
Dr. Dubey, you have 4 minutes left.
Yeah. So this this is for patients also. We could see expression of MIT-F, and MAP kinase pathway is regulating this expression. This MIT-F levels. So we found that triptandrin is downregulating downregulating BRAF and MIT-FM, but ERK and the MEK are increasing both in the nuclear and cytoplasmic levels. And when to confirm that we inhibited this MAP kinase pathway with UC-RO, so this upregulation was stopped and the MIT-F level was unaltered. So confirming the role of the ERK pathway. So usually when ERK is upregulated, the phosphatase MKP3 is maintaining the levels. But here we found that triptandrin does not interact with the ERK. So there are reports that MKP3 mediated dephosphorylation of ERK is dependent on the kinase CK2 alpha. So we checked whether triptandrin can inhibit CK2 alpha both by in silico studies, by colorimetric studies, and by Western blot. We found that it is a better inhibitor of CK2 alpha than the standard inhibitor CPA. And so we found that it is inhibiting this very good inhibitor of CK2 alpha. So the mechanism is it is downregulating the MITF levels. It is enzyme. It is inhibiting the two enzymes CK2 alpha and the BRAF. So the compound is a very good candidate to be studied against malignant melanoma. So this is the recently published work on this. A part of this work is published in the CMLS, and these are the previous report. This is the non-melanoma work. I did not present it here. So this is my lab. Thank you.
Thank you, Dr. Ruby. It's a good piece of work. I'll start with one question first, and then I'll ask others.
Yeah.
So triptandrin, that compound, when you treated A375 or B16F10 or A375,
yeah,
it is suppressing that mutated BRAF expression.
Yes.
What exactly does it? Have you checked that once that expression going down, the status of pattern of mutation also changing? Is there any DNA repair or any other
that
events happening?
We did not check. Only the expression is completely gone.
But whether that like a V600E, that position still exists, or is it changing the pattern of?
We did not see that.
And what happened in case of NRAS?
The mutation, we are getting a downregulation of all the BRAF. No, the NRAS we did not see.
Okay. Any other question, anybody? Yeah.
Thank you. It's a very nice, informative presentation. I have not a question, it's a query that you have used a mice that is bulbsy, right?
No.
What mice?
NOD skid mice. Which one?
This you have generated orthotopic
No,
model.
yeah, orthotopic skid mice, NOD skid.
So it's a immunocompromised mice.
Yes.
Okay, then thank you.
Yeah, the other one, for metastasis model, we used both. Metastasis model, we used the black cyst mice. That is immune. It is.
That is normal. That is normal.
That is normal.
So it's a human cell you have used.
That is not human. That is mice model.
For B16.
So both models we used.
For B16F10, he has used bulbsy.
Yeah.
And
no, not bulbsy. Black cyst.
Black cyst.
Yeah.
C57BJ.
That is mouse. B16F10 is a mouse melanoma.
Yeah.
And the other also, we used the other.
And
model.
A375, she has used skid mice.
Yes. A375 we used.
Any other question, anybody? If not, then thank you very much, Dr. Ruby.
Thank you.
It's a good piece of work. Thank you. Dr. Ajay, you can go.
So, hello. Our next speaker is Professor Satwinderjit Kaur, an eminent scientist from Department of Botanical and Environment Sciences, Guru Nanak Dev University, Amritsar. Her areas of interest are plant sciences, cancer, chemo prevention, genetic toxicology, natural plant products, etc. And she was the head of the department and chairperson of the member of board of control, then chairperson, member of board of studies. And she was also a faculty advisor. And she is the executive committee member of Environmental Mutagen Society of India. And she is a life member of Indian Association for Cancer Research. And she was awarded the DST Boy Cast Fellowship and the DBT Crest Fellowship. And she has a total publications of 136, citations more than 3,000, and H-index more than 30. So I would like to invite Professor Satwinderjit Kaur for her talk.
Thank you, Dr. Ajay, for the nice introduction.
So at the outside, I would like to thank the organizer, the patron, Professor Bharat Agarwal, and Alok and Anita, those who have, the organizers, those who have made great efforts to make this conference Incd 22 a great success. So we are running parallel sessions, so that speaks of the great success of this conference. So I had been associated with Incd 22 since its inception. So I'm here before you to just share with one of the research here. Again, there's a problem. So as now this, since yesterday we are all discussing about, so here I would just like to say the importance of cancer chemo prevention. So as the use of natural plant products, they play a key role. So it is better to prevent than control. So of course, once it is there, we have to do the steps. But as again, it's very slow. Yeah. So chemo, the treatment as such, the surgery and the chemotherapy, so it has many side effects that you can see, the fatigue, the toxicity, and the nausea and the hair loss. So here where so just to avoid, so nowadays we are also combining those chemotherapeutic, the medicine along with the natural products, and that has become more effective. So we worked on a plant, Onosoma bracteata. So it's running one slide ahead, slow. Yeah. So this is the plant bracteata. This is also called as Gauzuban, and it is at the northwestern Himalayas from it's there in Kashmir, and it is belonging to the family Boraginaceae. So it has this is our model plant. So this was not many studies have been conducted on this species. So here then we when we researched the literature, so it showed the Onsoma species as such showed many of the medicinal properties. It's showing anti-ulcer, anti-thrombotic, antibacterial, anti-inflammatory, wound healing properties. So we were interested just to pursue for this, and so it has many compounds on solvents, especially. Then we have others, naphthaquinones. So there. Yeah, so this is the extraction. We try to isolate various phytochemicals from this. So we had the extraction by in the order of the increasing polarity. So we got extracted with ethanol, then hexane, and then we went over to the last fraction with butanol, and then we had the remaining aqueous fraction. So then we were testing the various properties, how it is there. So all the fractions, they were tested with the, so we were doing some antioxidant studies. So out of the list of all these, we've just found here, which I've highlighted here, this is the ethyl acetate fraction, which is showing the IC50. So it is potent over here. So we took this fraction further, this Obia fraction. Further, there's another study, the anti-mutagen. We wanted also to see, because mutations are related to the cancer. So it's one of the consequences. So we did some plasmid nicking assay on these extracts. So where we were clearly seeing that the Fenton reagent, so it is showing the genoprotective effect. So although the effect was varying, but all these fractions were showing the effect. So this is the OBR fraction. Now we were interested to further see what is in it. So then we performed the various phytochemical analysis of this. Yeah. So this is one of the anti-mutagenic studies where against we are using the Salmonella typhimurium, and we tested it against the direct acting mutagens NPD and also S9 dependent 2-minofluorine. So here we are also seeing the OBR fraction to be much more active than the others. This is another study in another tester strain. This is TA100 tester strain of Salmonella typhimurium, and it is base pair substitution mutation tester strain. The other was the frame shift. So here again we are seeing the more activity in OBR fraction. Now looking at the phenolic content and the total flavonoid content. So this speaks that this OBR fraction is rich in phenolics and flavonoids. So then we did HPLC, and we could recognize the presence of catechins in OBR fraction, and in here there was there's one of the fraction, the mother extract, that which is the obit ethanol. So it is having apicatechin. So the blue one down below. So this is the HPLC analysis of these fractions. Okay, then we concentrated on the OBR fraction. So then we went in for column chromatography. Yeah, so this is how we did it. So the fraction, we got many fractions from that hexane ethyl acetate, and this is the OB4 fraction which is there. And then it was further purified, and at the end we are getting OBD1 fraction, which is a pure one. And we did it, no. Yeah. So this is where we tested what this OBD1 is. So this is it's showing a single spot on the TLC, and then we did the NMR studies and the mass spectroscopic studies, and then we could get this molecule which is 1,2-benzene dicarboxylic acid bis 2-methyl propyl ester. So this is the one we got. And the single compound in here. So this is the first report from this plant, and we did some then further concentrated on this anti-proliferative studies. We used these 3 cell lines, and again we are finding some good observations where the OBR fraction worked best against MG60. This is osteosarcoma cell line. And the fraction which we isolated from OBR, that is OBD1. So it is again more potent, showing this GI50 value as 37.53 here. Then also it is showing selectivity index, a much more higher selectivity index in OBD1 against MG63. Now we went into for the mechanistic studies, how this is, so this is the morphological study where in the phase contrast microscope, this is the OBR fraction. We are running side by side. So different GI30, GI50, GI70. This is in comparison to the untreated cells. This is the OBD1, which was isolated at its GI50 concentration. Now, this is the DAPI, where it is showing some nuclear fragmentation and condensation under the confocal microscope. This is 40x. Now this is the OBD1. So clearly as 100x, we could see some the fragmentation and condensation here in this case. So same studies, OBA fraction, a different how it is. This is showing the rounding of the cells and the nuclear blebbing and formation of these apoptotic bodies under SAM. So this is the isolated OBD1 fraction under SAM. So clearly showing the formation of going for apoptosis. So this we also used this just to see. This is acridine origin ethane bromide staining, where we are seeing the going in for the apoptosis. Here, this is OBD1 fraction. We are seeing the early apoptosis. And this is again now where we are seeing the MMP disruption. This rhodamine 1 to 3, it is leaching out when the cells are going in for apoptosis. This is with OBD1, the isolated fraction from this extract. This is in MG63 cells. Some flow cytometric studies were also done. So wherein we are finding the ROS generation with the increase in the concentrations. This is released and it's moving. So this is again ROS detection. We could see out in OBD1 fraction. Again, this the MMP potential. There is a disruption of the mitochondrial membrane disruption, and here we are showing the M1, which is the normal cells with the disruption, and M2 are the intact cells. So we are seeing it there increase. This is with the OBD1. Now here the cells were found to be arrested in G0, G1 phase. So you can see very clearly the cells are being arrested, the big peak, and OBD1. Then we also did an XN5 FITC PI staining, where we are seeing the cells in the apoptosis going in for, you can see in all these quadrats with the increase, they are moving towards the apoptosis. This is with OBD1. So we also did cell migration. So this is with the OBA fraction. So you can very clearly see the inhibition of migration of MG63 cells. This is with the OBD1. Then we did RT-qPCR. This is the OBA fraction from which we isolated OBD1. So here we are seeing the enhancement of the P53 and decrease of BCL2, mortalin, and also the decrease in cyclin E, CDK2. So OBD1, we could see very well the enhancement of the caspases. We hear caspase 3. So decrease of the anti-apoptotic proteins, and also there is a decrease in cyclin E and CDK2. So Western blotting studies were also done. So this is with the OBA fraction. So we could see very clearly enhancement in P53, caspases, decrease in BCLXL,
And these anti-inflammatory proteins, there were decrease in PAKT and enhancement of GSK3 beta. So this is with the OBD1. Very clearly, they are anti-inflammatory and they're also moving towards the apoptosis. So this is the paper which was published in Environmental Science and Pollution Research. And then the compound that we isolated. So this was published in Molecules very recently. So then also we did some hepatoprotective activity studies. Now this time we are doing in obit fraction. That was the mother extract from which all those fractions were isolated, and it's rich in apicatechin. The more is the concentration. So this is we did it on male visitor rats, and this was against the hepatotoxicity being induced by carbon tetrachloride.
Professor Kaur, can you wind up in 2 minutes?
Yeah, okay. So this is the study, and we could see some the serum parameters being modulated, and there were the modulation of oxidative stress parameters. So we did some this histopathological studies. They were showing clear indications of the protective effect of the obia from this Onosoma dactyata. This is where we had just seen about the cyclin D and P53, and we could see the modulation of these proteins. We did some Western also here. So this was published in Frontiers in Pharmacology. So this is the mechanism we proposed for this. And this is my team, which is working with me, and 4 of them are here, 5 of them rather are here. So this work which I'm presenting here is of Ajay Kumar. So he recently has finished with his PhD and is working with the Punjab Incubation Biotechnological Incubator, and it's doing good work. Thank you very much for your kind intention. Thank you.
Thank you, Professor Kaur, for your wonderful talk. So we can have 2 quick questions. Anybody? Yes, Professor.
So
next talk. Okay, fine. So I like the name of your extract, obia. Maybe you can use later as when you commercialize that name, obia. Yeah.
Thank you.
Our next speaker, Dr. Sunil Manna. He is scientist 7, Center for DNA Fingerprinting and Diagnostics, CDFD, Hyderabad. And Dr. Sunil did his PhD from IISc Kolkata, postdoc from MD Anderson Cancer Center. He is also top 2% biological scientist according to Stanford University ranking, recipient of Scopas Young Scientist Award, Fellow of National Academy of Science, and recipient of National Bioscience Award. Area of interest, molecular events of inflammatory disease and possible therapeutic intervention. And Dr. Sunil is going to talk on the role of advanced glycation end product in exerting adverse effect and its regulation. Dr. Sunil, please.
Fine. Good afternoon. First of all, I thank the organizer, especially Professor Bhartia and Anju. Of course, the overseas Professor Bharat Agarwal is not there. So my topics of talk actually role of advanced glycation in products in exerting adverse effects and its regulation. So I don't know how many people you know the age, advanced glycation end product. So basically age, it is just, when you are taking a lot of sugars or in the diabetic condition or aged person where metabolic activity is not that much, you cannot break that. So high level of glucose, normal system actually it becomes tri-carbon sugar, and these are like a methyl glycol, one of them. So these are sugar derivatives, and this non-enzymatically entangle with the protein or fat. Okay, so that actually results the formation of the whatever the products, either either protein or fats, that becomes named as a advanced glycation end products. So we are looking actually this normally in the body that have formed like that way, but in case of when you are taking the food, especially this the browning effect of the food happens because of this. So mostly the reaction happened like Amadori reaction, chips base, Maillard reaction, means that is the browning effect. That actually makes the basically you are barbecue the products that become brown. So there are a lot of actually advanced glycation in products would be there. So that's why careful to take those actually products, those who have the diabetic or the aged persons. So here actually a lot of listed products are there. So whatever the 3-carbon sugar moieties that used to entangle in the protein, I used to tell mostly. So here the structure of those, just going to that. And most actually damaging effect happen when, see, if it is a short-lived proteins, they used to degrade and just bypass from the body. But if it is a long-lived protein like lens crystalline, or a lot of proteins in the heart like tendons, ligaments, and cartilage, collagens. So there, that time lot of effects would be there. So normally actually when this kind of collagens becomes glycated, that time the tissue becomes rigid. So especially in the heart muscle, it cannot move that way. That way you will get the effect. So here a lot of actually listed diseases are there that are involved through the age. And age actually acts through its specific receptor called RAGE, receptor for advanced glycation end products. And it has different forms listed here, and which are the cytoplasmic domain. So if the cytoplasmic domain, then it used to do the signaling cascade, it used to follow the signaling cascade. And if it is just, you don't have the cytoplasmic trail, then they just becomes like a soluble. Lot of times this soluble age used to act as a therapeutics, but somehow it did not work as a therapeutics for other reason. But normally we are interested where these C-terminal domains are there, where a lot of signal transduction happen, and that leads to several deleterious effects. And I will come one by one those effects, how it is doing, how we can regulate. So these are the actually different cell signaling cascade already known, and mostly it is doing the, it induces reactive oxygen that leads to stress responsive transcription factor activation like NF-kappa B, and other sub pathways are also there like reactive oxygen like INOS also activating. So this is known reports. So for the research purpose, we prepared the age using the human serum albumin. Human serum albumin is inert protein which have no effect except the tonicity in the blood. And we actually incorporated age by like same non-enzymatic. If you incubate the age with the say for 7 days, then it will make the HSA MGO. We used MGO as a tricarbon sugar actually for to make the age. And in biochemically we measure that how much molecules are entangled, like a 31% proteins are entangled with this methyl glycol. So whatever we are using, if it is 100 microgram of age or HSA-MG we are putting, basically that is 31%. Okay, and this is actually, if you incubate this, it will give you this browning effect. In this test tube actually it is showing, and which actually amino acids side groups it is entangled. It is just going through that. If you are interested, you can go through that. Okay, then we find out that we use different like methyl glycol, D-glucose, glycoldehyde, ribose. Then we find out to activate See, our actually activation is NF-kappa B activation we are looking, and we find out that methyl glycol main age is the most effective, which is shown by gel shift assay, NF-kappa B activation as well as reporter gene activation. And in terms of deleterious effect, we use the cell and find out that it is giving almost 32% cell death. See, it is not huge, but it is a very slow reacting, slow acting. That's why it is giving just 32%, but that is enough for the inducing the lot of neurodegenerative diseases, which is the age-old disease. And in terms of apoptosis is multiple, we proved like a caspase activation and other things. And if you look actually NF-kappa B activation, which have role actually we used to tell it has proliferative effect. But in case of proliferative retinopathy, there also it is it is cell proliferating. But in most of the cases you have to regulate actually this NF-kappa B to regulate this action. And we find out not only the NF-kappa B, it is also activating AP1, NF80, okay, and whatever actually NF-kappa activation by activating the kappa B alpha degradation. Because see here we want to look the how it is doing the apoptosis. Okay, so to know the mechanism, we check this transcription factors activation. But here actually AP1, NF-kappa B, NF80 activating, NF-kappa B is known. Then we looked actually whatever dependent gene for these transcription factors. But also we got it increases the different proteolytic enzyme activation, because as it is a receptor ligand mediated action, so it is inducing some of these proteases, myeloperoxidase alkaline, that means it is helping in the degranulation. So when receptor ligand interaction, so it induces degranulation. And degranulation in that product, we find out interleukin 8 actually, it increases the level, which is shown in the supernatant and which reverse effect in the palate. So that means supernatant IL-8 is secreting. Okay, so this because of the IL-8, and we find out that the reactive oxygen other actually activation is going on, and antioxidant what it is reported, it is a acts through activation of the reactive oxygen. That's why we use several antioxidant that is blocking. Okay, that reactive oxygen, this is known fact. So and as calcium is increasing, as actually interleukin is increasing the level by degranulation, we find out it increases the intercellular calcium level, which is shown by the in this actually figure, that age increases calcium level, and because of the calcium, it also increases the calcineurin, one of the phosphatase activation, and I don't know, it is not shown from here. From here I can see. And it also increases NF80, the transcription factors which remains in the cytoplasms in the phosphorylated forms, and when it is dephosphorylated, it is to translocate to the nucleus. So now this NF80 dependent luciferase gene activation, as well as we also looked actually several other like gel shift assay and other way, and NF80 when it translocate to the nucleus because of the dephosphorylation by calcineurin, so NF80 dependent gene is the Fas ligand. This Fas ligand expression you can see as per the Western blot. Okay, and Fas ligand dependent luciferase gene expression. So like that way. So age-age interaction leads to intercellular calcium release, followed by calcineurin activation. NF80 phospho becomes non-phospho. That's why translocate to nucleus, and dependent gene Fas ligand expression. And if you put that here, actually we used anti-IL-8 antibody, anti-TNF antibody, but find out that anti-IL-8 antibody, if you use, that is actually that basically blocks this effect. Okay, in this gel shift assay, whatever actually see, if you block the calcium or anti-IL-8 antibody, so whatever this NF-kappa B activation by age is blocked, and the Fas ligand expression again, why it is also blocked by either blocking the calcium beta-2n, the chelating agent, or by anti-IL-8 antibody, but not the TNF antibody. TNF also very strong inducer of NF-kappa B, that is not affected, but IL-8 dependent action is affected. So that's why we are actually, and here it is also telling that same thing, the apoptosis assay. So in this actually cartoon, whatever we are telling, age-age interaction leads to the degranulation of the several things along with that, like a proteolytic enzyme as well as the IL-8. That IL-8 increases the intracellular calcium, and that calcium leads to the calcineurin activity, inhibited translocation, followed by the fast ligand. That's why it is inducing cell death, especially the neuronal cell death. And age increases reactive oxygen because some actually review us either it is increasing the reactive oxygen in our system is not. That's why we use this one. And I'm just passing. And now actually another deleterious effect like a diabetic response. So normally diabetic response is that, see, a lot of glutathione compounds which is activating the PPR gamma, that is acts as a anti-diabetic drug. Normally what it is doing, this troglytathione increases the PPR gamma DNA binding activity, and if you incubate with the age followed by troglytathione induction, the PPR gamma downregulation happen actually because of the age, and this PPR gamma dependent AP1, AP2 also decrease, and here actually NF-kappa B and PPR gamma have the inverse role. Just I will pass that on. But here actually the important findings here. See, if you put antioxidant, this PPR gamma, okay, down regulation, basically antioxidant cannot protect except the mangiferin. That's why coming to the actually our point that antioxidant, mangiferin protects age-mediated PPR gamma down regulation, but not other the antioxidant. That means whatever antioxidant activity is not actually whatever may be shown by the mangiferin, but mangiferin is showing something other effect. Mangiferin is derived from a mango, that is also true. And to our surprise, when you incubate this mangiferin with the just your in a test tube with your nuclear extract, different concentration of nuclear extract, it is increasing the DNA binding as such in the test tube. That means in vitro. That means the compound directly having some, basically it is enhancing the binding of PPR gamma DNA binding activity. Okay, and it is actually in silico also, you just put that one, that PPR gamma and troglytathione and mangiferin binding. Okay, and whatever the free energy calculation also finding that it is also looks like a troglytathione, which is known inhibitors of this known activator of PPR gamma. Okay, so that's why you are telling maybe because just I did not put so many data. Actually, we already published those data. That's why actually it is increasing the diabetic response by mangiferin. Okay, and now I will go actually that age increases lipogenesis. That means age, advanced glycation in products in the diabetic condition where high level of they are always obese. Actually, obesity is to come to the picture. So how it is doing in terms of mechanism? It whatever actually age increases along with the concentration, it increases the lipogenesis, means the lipid droplets are forming here. It is shown in this actually staining. And
Dr. Sunil, you have 5 more minutes.
Okay. So whatever actually this basically the transcription factor SRBP, it is activating SRBP along with the other transcription factors. And antioxidant basically blocking this other transcription factor like AP1 and NF, but not the SRBP, but mangiferin is blocking this, the SRBP, see, induced by this age. That actually surprises how mangiferin is doing several effects. So I'll just pass quickly these things. So that's why maybe we are telling maybe mangiferin has the huge potent role to down-regulate the SRBP that leads to the decrease of the lipogenesis. And what we find out, this age-age interaction leads to the autophagic action. And this autophagy is a prolonged autophagy, what we are finding out. And prolonged means it is impaired autophagy as per our data, whatever I will come to that. So age basically increases the autophagy here. And this autophagy actually, that age increases autophagy and lipogenesis both, but when we use this glucose, which is known inducer of autophagy, that is normally it increases autophagy followed by the maturation leads to the autophagy coming down in this towards end, but not the in case of the age. That's why telling that prolonged autophagy, all biochemical marker and other things we already proved that one, it is that I am not showing here. So that it increases autophagy, and that is prolonged autophagy. Whatever in this cartoon we showed in this journal JBC, that age-age interaction leads to several signaling cascade. Actually, that although you have the evidence or data, but I did not bring that one. It increases several transcription factors along with the SRBP, and that leads to the increase actually lipogenesis. And this what we find out, this mangiferin has a huge role in different actually functions related to the age-mediated functions. So now, really we can push this mangiferin as a therapeutics, and we find out this mangiferin is a very good inducer or activates catalase activity, and here actually multiply we prove that on the catalase activity is going, and the well-known inhibitor, the catalase activator is amino triazole ATZ, and we find out that this mangiferin in silico, whatever we find out, the mangiferin and ATZ are working in two distinct sites. They have the activity. So together maybe that will be good for the catalase blocking that way. And so I'll pass this one. And binding energy, other things we already calculated for this. And this mangiferin actually, look with the structure of this mangiferin, it looks like that way. Mangiferin is different from the mango, that is I think already known. And this is the different structure, a lot of hydroxyl groups are there. And this structure, whatever it follows, if you go for therapeutics, you have to look actually several of these the property like molecular weight less than 500, lipophilicity and hydrogen bond, all those things actually, these are Lipinski's rule listed here. So that might actually it follow. That's why we are actually, pushing this compound as a therapeutics, as it is in age increases the diabetic response or obesity, impaired autophagy or neurodegeneration or aging like that way. So that's why we are thinking to move actually that way. And some of the preliminary or data already we have. I am not presenting that one, how it is, especially the obesity one we are working in vivo model. Maybe next I will present that one, although we don't have that much data right now, so I am not bringing that one. So now actually this advanced, already advanced glycation input has the role in inflammation, apoptosis, that is mostly neurodegeneration, impaired autophagy, that's why maybe doing the cell death, and diabetic response, and obesity, that is lipogenesis. Okay, and maybe mangiferin go for the therapeutic in this actions. So, and the Sarboraga or whatever the concept of the Indian herbals, maybe mangiferin might follow like that way. So that way I'll stop here. Just I will acknowledge the students last 10 years, whatever actually this work actually done by the students. These 5 students are worked actually in this language, and the Safi Sarda, who is still working in this angle. Basically, she is now working in the AKRB in the obesity model, and role of the age in that angle she is working. And I will stop maybe here.
Yeah. Thank you, Dr. Sunil. Any question? Yeah. Yeah, Dr. Sunil, it's a very nice presentation. I have one quick question. AGE, it can increase diabetic property as well as the lipogenesis. So how we have measured that AGE can increase the lipogenesis? Which model you have used, and what are the marker you have checked other than what is called lipid droplet staining?
So in the laboratory condition, in the cell-based assay or what?
Yeah, cell-based or maybe any animal model.
See, oil red staining, whatever I showed you, okay? That is mostly the staining for the lipid droplets there. Now we are measuring in the super resolution microscope how many droplets are there. That way, computerized actually it will tell how many.
Is there any lipogenesis marker you have checked?
So that's a lot of see, the SRBP, the transcription factor.
SRBP is for actually cholesterol pathway or maybe fatty acid pathway, but other like surface marker like CD36.
We did not look
like
those things.
leptin, adiponectin, that type of thing.
We did not look actually those markers, but only just inside cell whatever SRBP and its dependent gene like AP2, all those actually we already shown.
Thank you.
Any other question? Okay, Sharon.
Hi, Dr. Mona. Nice talk. I have a quick question. 3 hours to 5 hours, biologically, how you can say that is yes, and because as you see, like as the Western you have shown, yes, multiple, but some of them, that effect is gone, in fact, when you went to the 6 hours or whatever it is. So the fundamental question is, the whole idea is about the biological aging. So how you can say just, 3 and 6 hours of biological aging and how you can correlate with the, because whole thing is depending on that. So can you please comment?
So means you are telling which, the lipogenesis or which experiment?
No, the whole story. The point is, it is a 48 hours, 62, the few hours or the 3 days as I perceive it, because some of them, number one, the effect is gone. It doesn't matter. All the things they need not to match. But in spite of that, say biological aging, how you can correlate this?
Any support stimulus, immediately it will induce the support reactive, support TNF or any actually like that. Here we are using age, which is a little bit slow. Support TNF, it induces within minutes actually a lot of signaling, but it takes its dependent gene to activate support 12 hours. Support TNF induces NF kappa be 30 minutes to 1 hour started. But depending on you will get like a 12 hours or overnight like that way. Same way it is.
You are very true. That can happen with any kind, So, but the question is still the original question. My concern remains. What you said is true. That can be any kind of stimulus. Having said that, how you can correlate when you say, because whole story is about the aging, how you can correlate 3 days and the 6th days with reference to the age, because whole thing we are looking with the neurodegeneration and cancer,
so
right?
Receptor ligand interaction. So when it is action is there, cytoplasm, a lot of factors is recruited. Okay, this recruitment of the whatever factors followed by kinases coming to that milieu. Okay, some receptors are very slow. So when ligand binds, say for this.
I'm sorry, that is not my question. My question is very simple. 3 hours or 3 days or the 6 days, whatever it is, how you can.
Maybe we can discuss during the tea break.
No problem.
We'll have tea break soon. We are running late.
Molecules you can go for less time.
Yeah. Okay. Thank you very much, Dr. Sunil, for excellent presentation.
So before inviting the next speaker, I have a couple of announcements. This INCD conferences we organize under the umbrella of Society for Nutraceuticals and Chronic Diseases. So this society started 6 years ago, and we have organized, this is the 5th conference we are organizing. So I would like to encourage everybody to take the membership of this society. And the first thing. Second, there is a panel discussion today in this hall. After the panel discussion, we will be organizing the general body meeting of the society. So please attend this general body meeting. Thank you. Our next speaker is Professor Chansey Mandal, and he is from Department of Biochemistry, Central University of Rajasthan, Ajmer. And his areas of interest are understanding molecular mechanisms of dysregulated gene expressions and cellular signaling network associated with the cancer, exploring the role of various metabolic diseases, including diabetes, obesity, and hypercholesterolemia, environmental risk factor on the peculiar differentiation property of epithelial cancer cells. He received his PhD from IIT Kharagpur in biochemistry in 2005, and he did postdoctoral research at the University of Texas Health Science Center at San Antonio. And he is the editorial member of various journals like Scientific Reports, +1, BMC Cancer, Frontiers in Endocrinology, etc. So I would like to invite him for his talk, to deliver his talk.
So there is one announcement. We are going to have another group photograph. We had one yesterday, but there are many new people who have joined, and it will be a totally different group. So we can actually gather around after the panel discussion is over. So we'll gather around in the same foyer where we are actually having a INCD and Deepak or Rangoli is there. We'll please gather there. just immediately after the panel discussion. You all are welcome to be there. Thank you very much. Yeah, please continue. Yeah. Yeah, and there's another next announcement is, if anybody who is not aware, we have our cultural evening and dinner in this premises only, and there's no need to go to the zoology department. It's totally wet, and it was outdoor event, but now we are going to do it indoor. Thank you.
A very good afternoon to all of you. Before I start, I would like to give a special thank to organizing committee, especially to Professor Alok Chandra Bharati and our session moderator, Professor Gopal Kundo and Professor Ajay Kumar, and the other audience who are over here. Today, I will give a talk on the title, A New Drug Targets Dysregulated Cholesterol Metabolism for Prevention of Cancer Growth and Metastasis. So, before I start, I would like to just give a brief introduction. I know everyone is know about this. Still, there are many factors, especially the environmental factors, usually the pollution, toxin exposure, meat consumption, alcohol consumption, physical inactivity, smoking. These are the environmental factors which always modulate the cancer potential, cancer development, cancer metastasis. In parallel, this environmental factor can also modulate different body parameter, as for example, body weight, obesity, cholesterol, diabetes, family history. So these are the intrinsic parameters which can also directly or indirectly involved in cancer progression and metastasis. So today I'll be discussing how the important role of high cholesterol in cancer progression and metastasis. So in the beginning, in 2015, when I have started my independent career in Central University of Rajasthan, so I have started the work that the top 50 countries which have highest cancer mortality rate, I have just pointed in the world map, and we have noticed that the top 50 countries, they are situated in a particular zone. Similarly, when we have marked the least 50 countries which have lowest cancer mortality rate, their position is in different. So you can see that highest mortality rate country, they are situated from the Tropic of Cancer towards the North Pole or Tropic of Capricorn towards the South Pole. Whereas the country which have lowest cancer mortality rate, they are situated in between Tropic of Cancer and Tropic of Capricorn. Basically, they are nearby equatorial zone. So they have somehow geographically distinct. So later on, I have again marked the top 50 countries which have the highest average cholesterol level in their population, and you see that they are the top 50 countries which have highest average cholesterol. They are situated from Tropic of Cancer towards the North Pole and Tropic of Capricorn towards the South Pole. So this geographical situation actually mimics to the geographical situation of the top 50 countries having highest cancer mortality rate. Similarly, you can see over here, the least 50 countries having lowest average cholesterol, they are situated in between Tropic of Cancer and Tropic of Capricorn. It is almost similar to the country which have lowest cancer mortality rate. So from this data, you can guess that there could be a link between the cholesterol and cancer mortality. So in that way, we have started our research career as independent. So then we have read many papers and we have noticed that yes, the high cholesterol rich diet shows an increased risk of various cancer, including pancreatic cancer, colon cancer. Elevated serum cholesterol or LDL cholesterol may increase the risk of several cancer, including breast cancer. Even cancer cells watch their cholesterol level to increase their growth. So in this context, we have started our research and we have compared the cancer mortality rate and cholesterol level through statistical analysis. And we have noticed that the average cholesterol of a country has directly proportional to the cancer mortality rate of a country. So this is the correlation coefficient. And then we have again counted the cancer mortality rate of different anatomically specific cancer versus average total cholesterol. And we have noticed that for various type of cancer like lung cancer, bladder cancer, ovarian cancer, pancreas cancer, breast cancer, there are positive correlation between average cholesterol and the cancer mortality rate. However, we have not seen the positive correlation in case of liver and cervical cancer, rather it is a negative correlation. So then we started the research in clinical sample analysis, and we have noticed that the patient of cancer blood have high cholesterol level as compared to the patient of healthy blood. Similarly, the cancer tissue contain elevated cholesterol level as compared to the benign tissue. This is the breast cancer tissue. And then we have compared the cholesterol level between the low metastatic MCSF7 breast cancer cell and the high metastatic breast cancer MD-MB231 cells, and we have noticed that a highly metastatic MD-MB231 cells have the more cholesterol as compared to the low metastatic breast cancer MCSF7 cell. So these data indicated that somehow cholesterol is associated with the cancer progression. It can be also associated with the metastasis of the cancer. So then we have analyzed the cancer sample of breast cancer, malignant breast cancer, as well as the benign breast cancer, benign breast tissue, and we have marked. We have checked the different cholesterol regulatory genes like HMG-CoA reductase. It's a rate-limiting enzyme in cholesterol biosynthesis pathway. LDLR, it's a low-density lipoprotein receptor. It is also very important for cholesterol internalization from outside to inside the cells. And the SRBF1, it's a crucial transcription factor which regulates the cholesterol biosynthesis. So we have noticed that all these crucial cholesterol regulated markers are more in the malignant breast cancer tissues as compared to the benign breast tissue. Similarly, we compare the cholesterol regulatory genes between high metastatic MD-MB231 cells and the low metastatic MCSF7 cells, and we have noticed that the high metastatic MD-MB231 cells have higher level of HMG-CoA reductase, LDLR, and the SRBF1. So this data further showed that not the cholesterol is more in the cancer cells, even cholesterol regulatory pathway is more active in the cancer cells. Then we have again searched various gene, those are related to cholesterol pathway, and in that time we have noticed that there are a gene set that is RXRA, SOAT1, squalene epoxidase, and ACAT1. These are the 4 genes. Out of these 4 genes, 2 genes are always upregulated in the cancer and 2 genes are always downregulated in the cancer. We have analyzed this data through TCGA database, and we have made the survival curve by combining all 4 genes together, and we have noticed that when we combine 4 genes together, the survival curve is poor for all cancer. We have mentioned over here bladder cancer, breast cancer, head and neck cancer, kidney, renal cell cancer, liver cancer, as well as the lung cancer. So these data further suggested that these 4 genes could be a gene signature when the cholesterol associated tumor is there. So to validate this thing, we have analyzed the tumor samples and between actually the control and tumor, and then we noticed that the tumors, this is best tumor samples, and we noticed that the best tumor tissue have higher level of SOAT1 and squalene epoxidase, whereas the RXCR and ACAT1 is downregulated. So this experimental data satisfied that the 4 genes could be the gene signature when the cholesterol associated cancers is there.
So then we wanted to know whether this cholesterol pathway can block or not. So everybody knows that there are many drugs which can block the cholesterol pathway. Especially I have worked simvastatin, and we have already proved in animal model that simvastatin can prevent the cancer growth and metastasis in a nude mice model. But today I'll be discussing another new drug, that is ADA. It's actually the conjugation of dopamine and arachidonic acids. Dopamine and arachidonic acids, sometimes they conjugate in our body. That is, our name is NADA, N-acyl dopamine. And there are few papers that NADA can be anti-cancer, can show the anti-cancer potential. Actually, this is the work in collaboration with Russian group, and also one of the collaboration is Professor Gopal Kundu is over here. And so we have shown that this ADA can show the anti-cancer activity as well as the anti-migration EMT activity. And you can see over here some of the data I am mentioning over here, that ADA can prevent the cell migration, it can prevent the colony formation, it can also prevent the epithelial to mesenchymal transition, as well as that can prevent the soft agar formation. So ADA can prevent cell migration, stemness, as well as the epithelial to mesenchymal transition. And then we want to know whether because there are few reports that there is a link between ADA and cholesterol. Then we thought that whether the ADA can block the cholesterol level in cancer cells, or it can decrease so we have treated the cancer cells with ADA, and we have found that the cellular cholesterol level of MD-MB231 cells is decreased when cells are treated with ADA. And then we have treated the cells with ADA and ADA plus cholesterol. We have found that ADA inhibited cell migration, stemness property was increased when we have treated with ADA plus cholesterol. Means cholesterol treatment reversed back the ADA mediated inhibition of cell migration, colony formation, as well as the matrix metalloprotease activity. Then we want to know whether the ADA can prevent the cholesterol regulatory pathway. So we found that ADA treatment can inhibit the HMG-CoA reductase, LDLR and SRBP1, and also it can inhibit the SRBP1 protein level. So ADA can prevent the cholesterol pathway to decrease the cholesterol content in the breast cancer cells. So then we want to know whether the ADA can prevent the breast cancer induced bone metastasis. So in culture model, what we have done, we have used one osteoclast precursor cells, that is called raw 264.1 cells. That cells can do the osteoclast activity after a certain condition. So where we added the condition medium of breast cancer cells, we have noticed that the osteoclast activity is more, that is measured by TAP activity. The TAP activity is a traditional method to measure the osteoclast activity. Then we measure the TAP staining of osteoclast cells. It can show the multinucleated osteoclast cells, and we notice that breast cancer condition medium can increase the TAP activity of osteoclast precursor cells. So this is the indication that the breast cancer can increase the osteolytic metastasis, and to further verify, we measure some osteoclastic marker activity like the TAP, cathepsin, and Incd2022-Hall is the master regulator of osteoclast activity. We have noticed that condition medium can increase the TAP, cathepsin, and the master regulator transcription factor Incd2022-1. So then we thought whether the ADA can block the breast cancer induced osteoclast activity. We have noticed that the ADA can decrease the breast cancer induced TAP activity. And there are two crucial osteoclastic factor that is called CSF1 and rank ligand. One is cholinesterol factor, another is receptor activated NF-kappa ligand. These are the two crucial factors which can activate the osteoclast activity. These are also highly expressed in the cancer cells. So we have noticed that whether ADA can block the CSF1 and rank ligand level in breast cancer cells, and our data suggested that ADA can inhibit the CSF1 and rank ligand in breast cancer cells. So further to verify this thing, we have analyzed some tumor samples, and we have noticed that malignant breast tissue has a high level of CSF1 and rank ligand level as compared to the benign breast tissue. So these data suggested that the CSF rank ligand is more in the metastatic breast cancer, and ADA can prevent the CSF1 rank ligand activity to block the breast cancer induced bone metastasis. So this is a cartoon. You can see that the SRBP1 is a crucial transcription factor which can regulate the cholesterol level, and this cholesterol can increase the CSF1 and rank ligand, and this CSF1 and rank ligand actually can increase the NFATS1 level in osteoclast precursor cells. This NFATS1 is crucial for the osteoclast activity by releasing cathepsin K and matrix metalloprotease. And in this case, ADA can prevent the SRBP1, which can decrease the cholesterol level. As a result, breast cancer induced osteoclast activity can be inhibited by the treatment of ADA. So this is a first report that ADA can inhibit the cholesterol level, and it can also inhibit the breast cancer induced osteoclast activity in cell culture model. So before I ending, I would like to say about my collaborator, the Professor B. Benzugulov. He's from Russia. Actually, this is the Indo-Russia project, and he's the collaborator. He has given the compound ADA. Professor James Radosovich from University of Illinois, Professor Alonag, and Professor Gopal Kundu, and Dr. Lokendra Sharma from Sanjay Gandhi Postgraduate Institute of Medical Science, and Mahavir Panwar from BHU. So these are my collaborators. Anyhow, at the end, I have not done the work. Work is done by my students. 2 students are present over here. And currently I have 6 PhD students. These are my PhD students. And these are the students, they have moved from my lab. They have done the work in my lab. And finally, the funding support. I got the support from UGC, DBT, DST SHARP, DST RFBR, recently DST CRG, and ICMR. It is approved. We are waiting for fund. Okay. Thank you for your nice attention.
Thanks, Professor Mandal, for your excellent presentation. So if you have any questions, yes, Subhash.
So it's a nice presentation, Dr. Mandal. Overall, cholesterol is good for cancer cells. This is what you said, right?
Cholesterol is good for cancer cell health.
Cancer cells.
Yes.
So do you also check good cholesterol and bad cholesterol?
No, I checked only total cholesterol, because inside the cells, whatever I check, it is an intracellular cholesterol. So it is all are bad. It is not HDL or LDL, because it is the cholesterol present within the cells. So we don't need to see HDL or LDL where we see the cholesterol within the cells. Okay.
Yeah, Professor Kundu.
Yeah, Dr. Chondi, good talk. So I have one question.
Yes, sir.
India, I have not seen, but outside, many people, they have established osteolytic bone metastasis model in rat or mice, especially in rat. Maybe it will be good if you can develop that model and check the efficacy of ADA, what you have used, tested in that in vitro model.
Yes, sir. I will try to do it, because some problem in actually working in animals. So I have collaboration now in Sanjay Gandhi. They have the mice facility, and hopefully we'll do over there. Yeah. Thank you, sir.
Yeah. Professor Anil.
Yeah, over here. I'm sorry, I may have missed it, but what is the evidence saying that cholesterol will induce RANKL?
Cholesterol can reduce the rank ligand?
Yeah, it is.
There is no much evidence, but that is the one we are going to say that when the cholesterol level is more, the rank level is more. But if you treat the cholesterol level, I published the paper in 2017. You can see that when we have treated the cells with simvastatin as well as the MBCD, methyl beta cyclodextrin, that is the cholesterol depleter, as well as the cholesterol lowering drug, we have noticed that the rank ligand level as well as the CSF1 level is down regulated.
But just a small comment.
Yes.
The correlation between cholesterol and cancer incidence, I completely agree. But could it be possible that cholesterol itself is not the culprit, but because of the activated cholesterol metabolic pathway, that converts a lot of chemicals that we internalize through.
Yeah, actually I understand. There are few papers that they are saying the 27 oxy cholesterol that could be the potential than the regular cholesterol, but we have noticed that cholesterol can upregulate ARC pathway, ERK1 by 2, that is ARC pathway, and that pathway can be one of the important factor for increasing the, of course, only cholesterol may not do, but one thing that when cholesterol level is more in the cell surface or plasma, that may inhibit the what is called permeability of many molecules and even the oxygen permeability. That may itself cause the hypoxic condition that may increase the cancer potential. That is my hypothesis, even though I have not tested yet.
Dr. Mandal, I have a question.
You work in cholesterol, so why are asking question?
Yeah, because your data and my data don't match at certain point.
But that's good. Then they will collaborate together, and how we can match it? Yeah.
So exactly. So we had worked on colorectal cancer, and we also did the panel of the cholesterol homeostatic enzymes. And what we found out that HMG-CR level is in fact inversely correlated with the survival. So if you have a higher HMG-CR, the patients are able to cope up better and survive better. Now here is a molecule which is inhibiting HMG-CR, and you are saying that that is the one which is beneficial.
Actually, what happened? Maybe if you analyze again the TCGA database, and we can search, okay, because there are so many cancer. Maybe colorectal cancer and breast cancer, it may be different. But everybody knows that the simvastatin is good for the cancer.
No, there is a controversy about that.
Yeah, there are few.
And that's where the personalized medicine come. And in fact, they talk about this, that the people who have high levels of HMG-CR do not need statins. So there is a question of personalized medicine.
So in that,
analyze the data.
Yeah, in that case, I can tell one.
Anyway, both
I can tell one hypothesis about that.
You
Okay,
can discuss during the tea time. Also.
Okay.
I have a good hypothesis. I'll tell you about that. Why the cholesterol is the simvastatin is not working all the time.
Okay, that's fine.
Thank you, Professor Mandal.
Thank you. Yeah.
Nice.
Thank you. Thank you, speakers. Thank you, chairs. And thank you, everyone, for the session. I would now like to request the chairpersons to please felicitate the speakers with a memento. Professor Satvinder Kaur. Professor Sunil Kumar Manna. Professor Chandi Mandal. And I would like to request all the speakers for a group photograph, please. Dr. Ruby, please join. Thank you, everyone. We break for tea now. We'll resume with the next session at 4:30 p.m. here. Thank you.
अगला अगला पीपीटी सर।